Apa? Nanti kita akan Tes, masuk. Bismillahirrahmanirrahim. Excellencies, distinguished guests, participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Peace be upon you. Good morning, and we are delighted to welcome to the event International Mini Symposium 2022 on Sustainable Agriculture and Technology. This event is organized in collaboration with UIN Wali Songo, Semarang, and Instiper, Yogyakarta. The Dean of Faculty of Science and Technology, UIN Sunan Kalijaga, Dr. Kurul Wardati, the Dean of Faculty of Science and Technology, UIN Wali Songo, Semarang, Dr. Ismail, the Dean of Faculty of Agriculture, Instiper, Yogyakarta, Dr. Dimas Dewara Puruhito, and also panel speakers, participants of Mini Symposium 2022. Before we begin, let me introduce myself. I am Aska Raisa Subagio. I will be Master of Ceremony for today's event. This event to bring together academia, lecturers, and researchers for delivering their experience of research and sharing the ideas on how to maintain the sustainability in agriculture and environment. Excellencies, participants, ladies and gentlemen, we are now going to listen to the opening speech delivered by Dean of Faculty of Science and Technology, UIN Sunan Kalijaga, and officially open the event. Dr. Khurul Wardati, time is yours.
Terima kasih, Mas MC. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wa syukrulillah wa la haula wa la quwata illa billah. Asyhadu an la ilaha illallah wa asyhadu anna Muhammadan abduhu wa rasuluhu la nabiyya ba'da. Allahumma shalli wa sallim ala habibina wa maulana Muhammad wa ala alihi wa ashabihi ajma'in. Yang saya hormati Bapak Dr. Dimas dari Instiper, yang saya hormati Bapak Dr. Yai Ismail, Dekan Fakultas Sains dan Teknologi UIN Wali Songo, Semarang. Yang pertama saya mengucapkan terima kasih atas kerjasamanya yang sudah kita bangun selama ini. Selanjutnya, welcome Dr. Hiroshi Arai and Ms. Elias to this event. Thank you for your willingness to provide your time to deliver your expertise on your specific topic. We are grateful for time and effort you
tidak masuk ke Indonesia.
I'm sorry, Bu Dekan, yours ke mute suaranya.
Dari tadi kah, Mas? Enggak.
Sudah.
Oh ya. Ya, saya ulang terima kasih kembali kepada Ibu Fariha Wilisiani, MPiotek PhD, Mbak Eka Sulistiawati dari UIN Sunan Kalijaga, dan Mbak Dian Triastari Armanda ya dari UIN Wali Songo, dan Bapak Dr. Erik Firmansah MSc dari Fakultas Pertanian Institut Yogyakarta. Terima kasih atas inisiasi dari kegiatan hasil kolaborasi dari 3 institusi, UIN Sunan Kalijaga, UIN Semarang, dan Institut Yogyakarta, dengan kegiatan internasional mini Symposium 2022 on Sustainable Agriculture and Environment. Kegiatan ini tidak sekedar kuliah umum, saya kira, karena kita bisa menghadirkan berbagai narasumber ya dari dalam dan luar negeri, khususnya dari Jepang dan dari mana ini ya, Alice Jack dari
Rusia.
Dari Rusia, ya. Thank you very much for Hiroshi Arai, PhD, and Alice Jack. Kegiatan ini ya punya tujuan umum ya, memang sebagai forum diskusi ya, forum sharing ide dan pengetahuan keilmuan bidang lingkungan dan agriculture. Kemudian di UIN Semarang ada program studi lingkungan, saya kira di tempat kami ada biologi dan konsentrasinya salah satunya adalah juga lingkungan. Kita sudah melaksanakan kerjasama, dan ini adalah sebagai salah satu implementasinya. Kemudian selain itu juga bisa meningkatkan ya kesadaran pentingnya menjaga lingkungan ya dalam rangka tercapainya sustainability bidang pertanian dan lingkungan. Dan juga supaya kita menyadari ya bahayanya kalau ketika kita memanfaatkan senyawa-senyawa kimia di lingkungan ya yang dapat merusak area pertanian. Kegiatan ini meskipun target utamanya para mahasiswa ya, khususnya mahasiswa di 3 perguruan tinggi tersebut, tapi alhamdulillah sekali, terima kasih atas partisipasi dari para praktisi, dari akademisi. Praktisi kami melihat dari BRIN misalkan saja, juga dari Dinas Pertanian, akademisi saya kira ada dari UII, juga ada dari Universitas di Kendari ya, Universitas Haluoleo. Terima kasih sekali atas partisipasinya ya. Mudah-mudahan kegiatan mini simposium ini punya impact yang besar kepada institusi, khususnya yang sudah melaksanakan kerjasama, dan umumnya kepada semua peserta. Juga bisa merupakan sharing ide barangkali untuk mengembangkan penelitian maupun ke pengabdian masyarakat. Harapan kita bahwa penelitian juga antar institusi ini perlu kita kembangkan, perlu kita galakkan, dan akhirnya ya bahwa perguruan tinggi ini bisa berkolaborasi baik dengan pemerintah maupun dengan masyarakat maupun dengan industri. Jadi banyak hal kegiatan yang kelihatan sederhana ini, tapi impact-nya saya kira luar biasa. Demikian yang dapat saya sampaikan. Mohon nanti barangkali dari Bapak Dimas ya dari Instiper juga dapat memberikan sambutannya, dan saya selaku mungkin host ya dari kegiatan di prodi kami, prodi biologi Fakultas Sains dan Teknologi UIN Sunan Kalijaga ini, kegiatan ini ya kita buka bersama dengan membaca basmalah, dan bagi yang beragama lain barangkali dapat menyesuaikan. Dengan memohon ridho Allah, kegiatan internasional mini simposium ini kita buka bersama. Bismillahirrahmanirrahim. Wa'alaikum minkum. Assalamualaikum warahmatullahi wabarakatuh. Saya kembalikan kepada pembawa acara.
Thank you very much, Ibu Khurul Wardati. We are now continue our meeting to the next agenda panel session. But before we move to that agenda, I would like to kindly ask all participants to turn on your camera. Yeah. I will take the photo of you. So please turn on your camera, please. Okay, ready? Okay, thank you very much. For panel session, there is 2 panel sessions. Session 1 will be moderated by Ibu Dian Aruni Kumalawati. Ibu Dian, please, time is yours.
Okay, thank you. Aska, am I audible?
Yes, please.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable participants and speakers of mini symposium on sustainable agriculture and environment. Very good morning. So glad to be here. My name is Dian Aruni. I will be your moderator for the first session of this symposium. For the first session, we have 3 speakers that already joined in this Zoom meeting with us. We have Hiroshi Arai, PhD. Good morning, Mr. Hiroshi Arai.
Selamat pagi.
Selamat pagi.
Could you hear me?
And the second is Mrs. Fariha Wilisiani, Master of Biotechnology, PhD. Good morning, Mrs. Fariha.
Good morning,
Okay,
Mbak Dian.
thank you. Thank you, Mrs. Fariha. And the third is Mrs. Eka Sulistiawati, MA, MIWM. If I'm not mistaken, it stands for Master of Integrated Water Management. Good morning, Ms. Eka Sulistiawati.
Good morning.
Okay, thank you. And for this session, each speaker will talk for about 25 minutes. And for Dr. Hiroshi Arai, it's special. We give you 30 minutes for your talk. And after the speakers have presented their topics, we will have a discussion for around 10 minutes. All the audiences can ask questions by raising your hand during the discussion session, or you can also write the questions in the chat box and I will read them for you. Okay, now let's start the session. I will invite the first speaker, Mr. Hiroshi Arai, PhD. Mr. Hiroshi Arai is currently is a researcher at the National Agriculture and Food Research Organization, NARO, Tsukuba, Japan. Okay, Mr. Hiroshi Arai, the time is yours, 30 minutes from now, please.
Okay, so shall I share the slides?
Sorry?
Shall I share the slides? Slides for the PowerPoint?
Yes,
Okay.
You may share your slides.
Yeah. So could you see the slides?
Yes, it's already appeared in the screen.
Okay, so selamat pagi, and thank you for inviting me in this symposium. And nama saya Hiroshi Arai dari Jepang, and saya di sini, sekarang di sini, the National Agriculture and Food Research Organization, Tsukuba, Japan. And if you have any question, please send email to this address at any time. So my research topic, my title is about symbiosis between the selfish microorganisms and the insects. And first of all, this is my introduction. I'm Hiroshi Arai, and my interest is about insects, Ranga, and microbiomes. And this picture was taken in the Gunung Arfak in Manokwari, near to Manokwari, Irian Jaya. And I have traveled to Indonesia from Sumatra to Papua. This is because my first interest is about insects. So I have traveled to these places and watched how plenty in the Serangga in Indonesia. So this experience was connected to my research topics. I will show you in the later. And my second interest is about symbiosis. When I was a high school student, I was crazy about to cultivate bacteria in my house. And first that I just bought the squid in the supermarket and soaked this kind of squid in the salt water, and 8 hours later, the surface will be shown this kind of luminescence. This is actually emitted by the bacteria. So I cultivated this bacteria in my house for about 2 years. And this is my primary interest regarding the symbiosis. So actually that I brought this bacteria to my graduate university, Tokyo University of Agriculture and Technology, about 10 years ago. And I talked with the supervisors, and I decided to study the insect as well, insect and the microbes, their interactions, because I'm interested in insects and microbes. By the way, what is symbiosis? Actually, the symbiosis, this is a definition. The symbiosis is an interaction between two different organisms living in close physical associations. So this is a simple view of the symbiosis. That is more familiar with you. And this is a Pokemon. And this Pokemon is actually the host. And in his hand, there is a symbiont that is a tree. And the symbiont and host have a mutualism. The both have a good effect because symbiont might offer the energy for the host, and the host will offer the praise for the symbiont. However, there is some parasites in the tree. So that this kind of the small Pokemon is harmful for the symbiont, the tree. So this is a parasitism. However, the Pokemon will directly harmful the symbiont, but sometimes this is indirectly harmful for the host. So this kind of host symbiont parasite, this kind of triangle association is sometimes be referred as holobiont. So I will show you in the later, and this kind of barrier strategy of living organism included in the symbiotic association. So how about insects? Actually, the insect is one of the most abundant species group in the world, and about 60% of all animal species are insects. And insects also have a various endosymbionts. For instance, there is some kind of essential bacteria in the aphid. For instance, the Buffonella is a name of the bacteria, and the Buffonella offers the nutrition to the aphid, and in vice versa, the aphid offers the nutrition to the Buffonella and the rooms for the bacteria. Actually, this room is referred as bacterial site. And when this Buffonella will be eliminated by using the tetracycline or as a kind of antibiotics, the aphid cannot live alone, cannot persist and generate offspring. So this kind of symbiosis is referred as essential symbiosis. However, there's many other kind of symbiont is already found in the insect. And those kind of endosymbionts are usually inherited from mother to her offspring. This is because the female generates eggs, which is plenty in the cytoplasm, so it is quite easy to enter into the eggs for microbes. However, the male will generate the sperm. This is lacked with plenty cytoplasm, so it is quite hard to enter in. So endosymbionts are hardly inherited from males. This is quite important point. Then I will show you my research topic, Obakir. And Obakir is belonging to the alpha protobacteria and quite near to the Rickettsia group, which is pathogenic bacteria of the animals and sometimes the vectored by mite. And Obakir is usually vertically transmitted from female ovaries to the next generation. And Obakir is estimated to infect about 40 to 60% of insect species. And the curious thing is Obakir is applied to the infectious disease control that I will show you later. And Obakir also manipulates the insect reproduction. And one of the Obakir phenotype is cytoplasmic incompatibility. In detail, as you may see, the non-infected male and female, of course, will generate a viable offspring. This is quite normal. And when Obakir is in the female and males are free for Obakir, the offspring will also be viable. By contrast, if the male are infected with Obakir and female are free for Obakir, the offspring will be killed by Obakir. This is a cytoplasmic incompatibility. However, when both male and female are infected with Obakir, the killing effect induced by Obakir will be rescued. So this complicated association is referred as cytoplasmic incompatibility, CI. And Obakir-induced CI can eliminate uninfected individuals. So in Yogyakarta, already this kind of attempt is ongoing. In detail, a researcher inoculated Obakir in the Aedes aegypti, which vector the dengue virus, and he planned to release the Obakir-infected Aedes in the field. In Yogyakarta, the attempt started around the 2016, I suppose, and from that point, because of the CI phenotype, the number of Obakir-infected Aedes had drastically increased, and at the same time, the number of the patients for the dengue disease had decreased. So it is estimated that the Obakir could be effective to control dengue infection. So this kind of project is supported by the Melinda Gates project, and Obakir could be useful, but questions are still remaining. The first question is how Obakir manipulate insects. And second question is how Obakir have developed such selfish strategies. So I have so far worked on to resolve the questions. And my research target is male killing. This is a selfish strategy of symbiont such as Obakir. Then maybe some of you think why Obakir dislike males. This is one of the reason is it is quite simple because males are useless. Because the Obakir cannot be inherited from males. Obakir likes males. and Bobakir want to kill males, and Bobakir want to gain the number of the female. And second point is a resource reallocation. Please imagine that when the brother and sister are on the same table, if the milking happen, the brother will be killed maybe, and so the sister can consume the 2 dishes. So that is quite happy for her. So resource reallocation is adaptive for the sisters. So this kind of milking happen in everywhere, and female bass insect population can pass it over 1 century. For instance, in the Hyperymnosporina butterfly. And my research topic is this one, the Homona magnanima, belonging to the Lepidopteran insect Totruchida. And this insect is known as a serious tea pest in East Asia to Southeast Asia, and that damages the tea leaves. And Obakir was also found in this species. Based on our survey that from the Japanese population, we found 3 Obakir strains which do not induce a milking phenotype. We named the 3 strains as WHM A, B, and C. The phenotype for WHM A, the phenotype is still unknown. For B, WHM B induces cytoplasmic incompatibility CI that I showed before, and WHM C compensated the host fitness by by shortening the larval period and increasing the pupa weight. In contrast, from Taiwan, that I found a strain WHM named as WHMT, which induced the milking phenotype. So I firstly checked how the phylogeny of Obakir strains, and actually the WHM A-type strain and the B-type strain are in different grade. However, the WHM C-type strain and T-type strain couldn't be distinguished based on the Obakir, WSB genes, and MLSD typing genes. So please remember that WHMT induces milking phenotype, but WHMC do not. So why the C and T types are so closely related? So why the closely related Obakir strain exhibit different phenotype is my research, the first research theme. So to resolve the question, the aim of this study is to elucidate causative factor of milking and evolutionary processes of the milking Obakir. To answer this question, I have so far surveyed these 3 points. First of all, host effect, host genetic background is essential for Obakir phenotype. Please remember, WHMC are originally found from the Japanese population, but WHMT are found from the Taiwanese population. So I firstly saw that if WHMC are fixed to the Taiwanese genetic background, then the strain might show the milking phenotype. But the answer was no. The host genotype didn't affect Obakir phenotypes. And second point is bacterial density in the host insect. Actually, WHMC, I firstly saw that WHMC are as high density as WHMT. The strain can induce milking, but the answer was no. The WHMC didn't cause milking regardless of its titer. And so the third point is the pyrogeny. As you may know, that non-milking WHMC and WHMT are closely related. However, the genetic differences are still unknown, so I want to survey that point. To answer the genetic differences among the strains, I firstly purified the Obakir from the Obakir infected host line, WT12 and the Yakushima line. And by purifying the Obakir, extract the DNA and sequence by using the sequencer and assemble the genome by using canoe and constructed a complete genome of WHMT. And for WHMC, because Japanese population usually have 3 Obakir strains at the same time, so we have to separate in each. And by using single cell analysis, we purify the WHM A, B, and C, and sequence using NIE and MiSeq, and assemble by using the canoe. Then compare the WHMC and WHMT. And this is a simple view of the result. And this circle view indicates the WHMT genomes. And this is a part. The red color region is the WHMC, the commonly lacked region in the WHMC. So actually, this region was prophage doubler. The length is about 76 kilobits. So we will refer as 0076. So 0076 was always conserved in the American WT strain, but lacked in WC. So I have to introduce the life cycle of the bacteriophage. As you may know, that bacteriophage have to infect the bacterial cells, but in the lytic cycle, the bacteriophage will be replicated smoothly in the bacterial cell, and after they are too much in the bacterial cell, the bacterial cell will be collapsed. And this is a lytic cycle. However, some of the phage will be integrated in the bacterial genome. So until some stress, or such as environmental stress or temperature stress will be occurred, the bacteria can survive very easily. But if such kind of stress happen, the replication of the bacteriophage happens, so the bacterial cell will collapse. So bacteriophage usually have this kind of life cycle. And regarding my phage doubler, Obakir phage doubler, there's mainly the 4 characters. The first of all, the Obakir phage doubler is a dsDNA phage and existing Obakir genome as prophage embedded in the genome. And the curious point that is the double phage is always found, almost found in the almost all Obakir strains. About 80% of Obakir strains have a double phage. So please remember that the Obakir infect the insects, and insect is the most abundant species group in the world. So the Obakir phage doubler is also the most abundant viruses in this world. And the curious point is that the Obakir phage doubler influences Obakir phenotypes. Sorry, this background picture is the double particle purified from WHT. So to answer how the 0076 affect the Obakir phenotypes, we firstly annotated the genes on the region by using the software Blocker and Patrick software that we found that 83 genes on this region. And by using the RNA-seq analysis, 53 genes out of 83 genes confirmed to be expressed. And these genes includes several valence-associated gene effectors. For instance, WMK genes harboring the helix-turn-helix domain, and SIFP genes, which harbor the unkinin lipids which target the protein, and the other domain was papain-like cystinase proteinase domain. So as a background, this is a bit complicated. The WMK and SIFP and SIFA genes were found from the Obakir strain double male strain, which do not induce milking, but induce cytoplasmic incompatibility, CI. And please remember, the CI will be occurred when males are infected with Obakir and females are free for Obakir. And similarly, when SIFA and SIFP gene are in the male body and female are free for such kind of genes, the CI will happen. So actually the SIFA and the SIFB was a candidate, was a causative factor for the CI. And when female have a SIFA, the CI phenotype will be rescued. By contrast, when WMK gene were present in the bacteria, so WM gene will induce the defects to the host insect, and the WMK gene killed about 30% of all male individuals. So because these genes could be found in the double region of the WHT strain, so the question is, will 0076 genes induce milking in insects? To answer the question, we use this system, Drosophila melanogaster acting alpha system. Briefly, the acting alpha system will harbored by the female, and males harbor the US and the target genes. And when the males and females were mated, the offspring will be the only US and target gene harboring male and female, and both the acting alpha and US and target genes harboring female and male will be exist. So the both factors are in the same individual, the gene will be expressed. So I count as the number of males and the female. This is quite simple. And as a result, this is a simple view of the result. And first of all, the WMK1 and WMK3 reduce the viability of the males as well as the females. But all across adults were female. This was quite surprising for me. By contrast, WMK2, 4, and CFP gene didn't induce such kind of phenotype. So the curious point is that WMK1 and 2 and WMK3 and 4 show the different phenotypes but are adjacent, tandemly allied. So actually the CFA gene and CFP gene are tandemly allied. So there might be some functions among the interaction between the WMK and 1 or WMK3 and 4. To test the question, we simply co-expressed the WMK1 and 2 and 3 and 4. And the result was quite also surprising that when WMK increased, so the rescue have occurred. And similarly, but more surprisingly, when WMK3 and 4 were co-expressed, the number of female was drastically increased, but the number of males are still low. So this means that co-expression of WMK3 and 4 induced a strong male-king phenotype. And this is the first identification ever. And this is our laser model of the WMK in flies. And actually, the first thing is that males and females have a different susceptibility toward the WMK gene. And WMK1 and 3, such as a virulence WMK, could induce the strong virulence towards the insect. So males and females could be killed by the gene. However, the presence of WMK2 or 4 will reduce the virulence of the WMK1 and 3. We still do not know how the WMK4 or 2 will interact with WMK1 or 3, but we suppose the 2 or 4 have a suppressive function of the virulence. And this kind of difference in susceptibility and the interaction between the tandemly allied WMK gene might be related to the mechanism of the male-king function, the Obakir. And the last question is, how has Obakir acquired the 0076 and evolved male-king ability in tortoise, my target insect? To answer this question, I back to Indonesia. I surveyed the Obakir infection in Taiwan, Vietnam, and mainly in Indonesia. I stayed in Yogyakarta about 3 months in the 2018, about already 4 years ago, in the UGM, and collect and layer insects from 40 localities or 4 countries and detect Obakir and 0076 by PCR and NGS. This is a simple view of the Obakir infection in tortoise. And could you see that WMC are plenty in the tortoise that was collected in Japan, Taiwan, or Vietnam, or Indonesia? And actually, the 46 species out of 62 species were infected with WMC type strain. However, the virus 0076 was only found from Taiwanese Homona magnanima. And this is a sex ratio of the insect. Could you see that the red highlighted Homona magnanima only show the female birth sex ratio, but other insects do not show the female birth sex ratio. So male-king was confirmed to be observed in Taiwanese Homona species. So the important point is that to study the Obakir infection in Homona magnanima because the other related species do not show the male-king phenotype. So I checked the Obakir infection in Japan to Taiwan and in Taiwan, and we found that in here, from the mainland Japan to the Okinawa prefecture near to Taiwan, W A-type strain, A-type strain, and B-type strain, and C-type strain. These three type strain are the widely conserved. By contrast, in Taiwan, the W A-type strain and B-type strain couldn't be identified. So the all identified Obakir was W-T strain. And based on our detection of W76 in this region, this is quite simple that in Japan, we couldn't identify W76 harboring H. magnanima. But in Taiwan, the W76 could be identified. So W76 was only identified from Taiwan. And this is the simple summary of the evolutionary process of Obakir. First of all, W A, B, and C-type strain were conserved in the ancestor tortoise species, and the three strain was descendant to the current species. And some of the strain are conserving the tripleted Obakir, but some of the strain like W A or B strain and are now in the type C. So such kind of things happen in the ancestor of Homona magnanima. Maybe at the specific point, the ancestor of Japanese population and the Taiwanese population was diverged. And the ancestor of the Japanese population harbored A and B and C type. But the ancestor of the Taiwanese H. magnanima only harbored W&C. And at the same time, at some certain point, the W76 infect the W&C and came to be the W&T, which induced a making phenotype. However, please remember that W&B actually induced a cytoplasmic incompatibility CI. So if the W&ABC infected males are mated with ABC infected female, their offspring will be viable. However, if ABC are infected with male, that's mate to the female of only W&T harboring, which harbored only W&T, the offspring will be lethal. So geographical barrier should be needed to maintain the W&T infected individual. So this precedes this view because Japan is an island country as like the Indonesia, and Taiwan is also isolated from the mainland Japan. So this kind of the boundaries are quite important to maintain the W&T in Taiwan, and that was not identified in Japan because if the W&T are in Japan, that will be eliminated by the function of W&A and B and C. So W&T may have acquired making in Taiwan or mainland inland in China that was descendant from the phage might infect the W&C strain that was descendant from the ancestor tortoise species. So my speech overview is there is a relationship, the phage tableau, Obakir, and insects. And what we found is that phage tableau is a fixer, and Obakir is just manipulated to induce making phenotype, and insects are just beaten. And this is a summary and insights. I have answered first question was what is the causative factor of making, and my answer is phage tableau is a license to kill males. And second question was how Obakir acquired making phenotype, and my answer is W&T acquired making ability in Homona magnanima. And this work was supported by those members and in Japan and other countries. And thank you for listening. If you have any question, please ask me. Thank you.
Okay, thank you very much for Mr. Hiroshi Arai for the presentation. And honestly, it also brings a lot of memory for me because I used to work for Eliminate Dengue project that now transformed into World Mosquito Program. Okay, and for the audience, please hold your question because we will move to the next presenter. I will invite Mrs. Fariha Wilisiani, PhD.
Okay. Can you hear my voice?
Yes, very clear.
Okay.
And
thank you.
Mrs. Fariha Wilisiani currently work as a lecturer at Agrotechnology Agriculture Faculty in Ispo, Yogyakarta. And without any further ado, the time is yours. Please, Mrs. Fariha.
Okay, thank you for the time given to me. This is I share my PPT is clearly appear. Check first. Test. Test. Hello. Test. Hello, Mrs. Dian.
Yes, I can hear you. Please continue.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. The Honorable Dean of the Faculty of Biology, Science, wait a minute, of the UIN Sunan Kalijaga, and the Dean Faculty of the Biology from the UIN Walis. And also Mrs. Dian, thank you very much for the opportunity given to me. I think this is the nice moment for our collaboration between three universities among us, and in this event, maybe the good starting for sharing of our knowledge, sharing of the information of correlated with the theme and topic in this event, with the topic of the sustainable agriculture and environment. Recently, this topic is, this is very sexy because so many discussion and also sharing knowledge from the so many kind of aspects. But in this time, maybe still correlated with the Araisan. Good morning, Araisan. Thank you for your sharing of your knowledge about the insect. And in this time, little bit difference, but for sustainable agriculture, that the topic from this event. Test. Maybe any back sound? Test. Hello.
Okay, we'll try to cope with this. For the host, please mute one of the audience. Okay, thank you.
Okay.
Please continue.
Just be focused. Okay.
Okay.
So, with the correction of this topic about the sustainable agriculture and environment, maybe Araisan in the first is describe about how to mechanism about the insect, but in this time I will maybe just little knowledge of my little knowledge which can be shared together in here is about the plant virus diseases and also correlated with the insect mechanism. And I can say maybe this is a very good discussion after that. With the title Emerging Plant Virus Diseases of Bicomovirus, which is the one kind of the biggest of the plant virus diseases on the world. I will focus on the discussion about the from metagenomic analysis for rapid detection methods. So before I describe detail about this theme or topic, maybe I want to introduce myself. My name is Fariha Wilisiani. You can call me Fafa. I am the lecturer and researcher at Agrotechnology Faculty of Agriculture in Ispo, Yogyakarta. So maybe in our campus, this is also become the one of the program that also concentrate to pest and disease management. But our campus is also focusing on the plantation and forestry. In here, I will describe detail about the major section of the plant virus diseases, especially in the one group. As we know that for sustainable agriculture, plant virus diseases are the most threat leading to economic value of crop losses. We know well that some of our farmers have the huge of the lands, crop lands, but some of the and I think the major of the problems of our farmers is still cannot manage well about the crop losses, especially correlated with the plant diseases. And many viruses infecting of the several crops has been described in the literature. I think so many literature you can explore more. However, some of them are become the emerging frequently through the outbreaks. I think the definition or concept of the emerging plant viruses is very widely, but in this time I will focusing on 2 points that the emerging plant viruses is maybe it can be the new one. It's mean not previously known. I means that the plant viruses is become the newly one in the world. So nobody is known well about this disease before. But also the emerging plant viruses also with the context of the viruses that maybe it can be non-viruses that change in the environment or ecosystem. Maybe we can focus especially in Indonesia. We have the tropical climate that our environment is also affected by so many kind of factors. So the emerging plant viruses can be effect for the new variant, and consequently is the spread rapidly expand into the new disease. The factors of the driving emergent plant viruses is so many kind. In here, I will describe just a few of them. The one is from the viruses. So the genetic variability of the virus, especially the plant virus, is very widely. We can check in the International Committee of the Taxonomy Viruses. Most of the plant viruses also has already described or identified, and the genetic of the genome of this virus is also very variable. So it occurs the diversity and the spreading rapidly of this virus. The point of the number 2 is the change in the agricultural practice. This is very related with our theme, sustainable agriculture, that of the climate change, or maybe global warming, or maybe in our tropical that this is rainy season or this is the summer season. It is not so clearly. So I think this is also very effect for the emerging of the plant viruses. Point number 3, sorry. This is the increases of the population correlated with the Araisan described before. Correlation about these viruses with the insect as a vector. So in Indonesia, as tropical country, we have also so many kind of insect. Maybe insect that described before is one also the major in Indonesia, and some of the insect have the role to be the vector of virus. It means that if some of the insect has already get the one kind of virus, so it's be easily to rapidly spread in this environment. So this has become the badly environment, badly condition for our farmers. And the last one is the long distance transport of plant materials. I means that in that time, the export-import activity is very easily to come and also export in our country. So the quarantine is the key point for controlling of this factor. About the emerging viruses, I will focus on the baculovirus because this is still become the most plant diseases severe, especially in our country, Indonesia. May I compare with Japan? So Araisan, I also study in Japan from 2015 until 2019. So when I compare with the condition in Japan, it's little different, because in there the environment is consist, is the system is very support. But in Indonesia, maybe our farmers just get the length of crop in the widely of the land without the system for like quarantine, or maybe like net or something like that. So the emergence of the bighorn virus, that this kind of this virus is also transmitted by the vector of insect, namely whitefly. I will describe you after that. And also can impact for the variety of the vegetables crops, not only in Indonesia, but also in the worldwide. It means that not only in the some of the group of the plant, but the host range of this variety is very wide. Some of the literature describe well about the infecting of bighorn virus in the dicotyledonous plant, especially not only in the economically important vegetables and ornament species, it's mean like flower or something like that, but also in the weeds. So it's become the most concern, I think, for our scientists and also the farmers on the field, that some of the weeds reported to be increased the severity of these viruses. And if we describe about the host range, this is very large. Solanaceae, Cucurbitaceae, Malvaceae, Euphorbiaceae, Fabaceae is also included in the host range of this bighorn virus. So infection of this bighorn virus were found and spread rapidly in Indonesia, especially. I have already reported, especially for Cucurbitaceae and Solanaceae plants. This is just for the example of the diseases that can occur because the bighorn virus infection with the mosaic and leaf curl. So in Indonesia, maybe some of our farmer is not so familiar with the if we describe bighorn virus, but if we say keriting, or maybe in Chile, especially the namely is the kutu kebul, or maybe yellowing in the appear of this plant. This is the most common symptoms of bighorn virus infection, like that. So the leaf is become yellowing, can totally or maybe just the spot of this leaf, and also become curly of this leaf. And for the wall of this plant is become stunting. It means that if the condition of the morphology of this plant is like that, so we can know that the mechanism of the physiologies and finally for the crop production is also become not well, I think. If we want to more know about the bighorn virus, this is the including the Geminiviridae family, with the characteristic is very, very, very small. I with my sensei ever try to use the electron microscope, and this is also very very complicated problem, because the size is also very small and become twin. So some of these bighorn virus is cannot be detected well, often by the microscope electron. And if we describe about the how about in the worldwide, so maybe I will minimize. So until now, the currently of the emerging of the viruses is increasingly, and from the first step, the scientist is categorized for the bighorn virus become two group. The old word, it means that the new reported in the early of this infection and become spreadly more badly in the worldwide, so become the new group. The new one is the new word. So we can know in this figure that from the one of the family of this virus, Bicovirus become the maybe 90% more the member among the Geminiviridae. Sorry. Okay, sorry, I little get a cough. Okay, and especially in Indonesia, we have already reported a new type of the Bicovirus. Namely tomato leaf carniodelhi virus or squash leaf curl virus. And most of the reported scientists in Indonesia is reported from the host of this virus from cucurbit and Solanaceae. Sorry, wait a minute. Okay, I will continue. In this worldwide, so the occurrence of the tomato leaf carniodelhi virus is also become the number one. Is it clearly my voice? Become the tomato leaf carniodelhi virus, including Indonesia. So from the report, Mizutani and Septariani, this is the first report of the tomato leaf carniodelhi virus, membering of the Bicovirus that infect cucumber in Indonesia. But some of the similar symptom is also not only occur in the cucumber, but also in Solanaceae. So I interest to explore more about these plant virus diseases. This is the common of the Bicovirus symptom with the vector of the Bicovirus. Some of this virus is also spread by mechanically, but especially for Bicovirus, the vector is become the key point of this spreading of this virus. So the size also very small, maybe just 0.8 millimeter. And until now, so many kind of group are identified kind of the vector, namely whitefly, Bemisia tabasi. We know that the biotype Q and biotype B is the most severe of this vector. This is a melon field in Bantul that I conducted the research in the 2015 in Indonesia, and I bring the sample to Japan for identified and molecular characteristic. So if we want to know about the molecular characteristic, why our research is also including the molecular, not only just for the symptom, because in Indonesia, some of the reported is just stuck in this, I miss the morphologically identification, but in the world, the kind of this Bicovirus reported is also including the molecular report, and this is become the very important information for the spreading of this virus. So this virus is have the two kind of group. The one is the namely bipartite. It means that the virus has the two kind of DNA in the one virus, but the another just the monopartite that have only one kind of this DNA. Why this is very differently? Because this molecular composition of this genome of the bipartite and also monopartite will affect the diversity, the infectivity, and also severity to the host strains of this plant. I will not describe detail about the molecular. Maybe some of the participant is also the undergraduate and not focusing on the molecular biology material, so just the overview of this genome. But if you want to get the more information about that, so please contact me directly. So this is the combination of the molecular genome for the Bicomovirus, and recently the new one of the molecular genome is also identified from the same virus of Bicomovirus, namely beta satellite. For the size, not so big, just the half from the major of the molecular genome, but this satellite is reported have the important point for infectivity of this virus. Some report show that the DNA beta, DNA alpha is more severe than the others. This is just the namely of the open reading frame. Maybe some of you know well about the molecular genome, that the open reading frame is very significant point of this part to regulate the protein of this virus. We know that the virus is very simple organism in the world, just has the molecular genome, and some of them also has the capsid or maybe coating protein, something like that. So it's very easily for it's mean the breeding to be more spread also, and especially also for Bicomovirus, this is very similar to RNA virus. We know from the some of reported that shown the data, the RNA virus is very easily to it means that the molecular genome is will be changed and something like that. So the Bicomovirus is also similar. The composition, the combination of the gene in this molecular between the bipartite, monopartite, or maybe also has the beta satellite molecule. This is very easily to, for example, recombination, mutation, and something like that. And it will be occur in the severity and effectivity. I want just tell about the common region, because this is the most important part of this genome, the DNA of the Bicomovirus. Why? Because the common region can be generated for the detection method. So if we has already known the symptom from the morphology of this plant, so we can continue to detection of this virus. This is proofly infected by Bicomovirus or not. So from this region, we can we can make some of the primer. Maybe in the PCR we need a pair of primer that can amplify this region. And why in this time I interest for analysis especially for metagenomic, because if we know the detailed information about the molecular component of the virus, so this is very easily to identify. I means that the most common of the methods for the metagenomic analysis is we use the PCR followed by sequencing like that, and we can get the full length of this molecule. Okay, I will continue. So after we get the PCR cloning, we can get the full length of this DNA with the pair of the primer. Because the Bacteriophage is very small of this genome, so we just need to pair of the primer that can sequence or identify full length of this genome. If we compare with the other plant viruses, maybe the size is maybe triple or maybe more bigger than this size. So it's mean that in the PCR cloning, we have just combined all of them.
Recording in progress.
And this is the similar for the DNAP. We just have the two kind of the pair of primer and can amplify full length of the genome. And currently the new methods, I mean Araisan also described before in this presentation. The method of the next generation sequencing or NGS has been developed. This is become the highly efficient and rapid DNA sequencing compared with the standard methods. NGS technologies using the Illumina and combined with the especially bioinformatic program were successfully described the population of the diversity of Bacteriophage in naturally infected plant. This is just the step. Maybe I also use these methods. So we use the molecular genome of our sample. We prepare the library preparation and use the NGS sequencing, and after that we can analyze our data.
Excuse me, Mbak Fariha, you
Yes.
have 5 more minutes for the presentation.
Okay. Thank you. And this is the result of my sample that had already analyzed by NGS. Okay, I will not very detail, but I just want to show that the result from the NGS sequencing and also PCR method is same with the same result. So how about the metagenomic analysis with the NGS? So we can know the virus species or strain already known to be present in the area, or maybe the new virus species or strain, or maybe this is completely novel. This is my result study in Japan. So directly, this is the Pyrogenie methods like Alison before described. So we can get the position of our virus with the known or reported viruses others. And in my first result, I get the recombination result from the two kind of viruses that become emerging in Cucurbitaceae in Indonesia. So in this data, I can describe that the virus is recombination result from tomato leaf curl Delhi virus and squash leaf curl China virus. And I also test the infectivity of this isolate, and the infectivity is become severe. This is the figure out the recombination event. Another report is that another samples showing the tripartite like genome. I described before that the Bacteriophage commonly just have two kind of DNA, DNA A and DNA B. But in the samples, I found that this virus has the tripartite like genome. So double of the DNA A and the one of the DNA B. I also check the infectivity of this isolate, and the infectivity, the host range is also rapidly increases. This is just the theoretically about how possibility of the Begomovirus and satellite evolution. Maybe because the limiting time, I will not describe detail. And from this possible scenario, the recombination and also the tripartite-like genome is logically can be occur in the naturally infected plant. And from this metagenomic analysis, I continue to develop the detection of Begomovirus that we know the ordinary methods that we can use for detection of Begomovirus is use the PCR method. Maybe this is very common in our laboratory methods, but I try to explore more about the rapid detection, how to detect the PCR rapidly, efficiently, and on-site detection. So we conducted the research with the methods of the loop-mediated isothermal amplification or LAM assay. Maybe the concept is a little bit different. For the step, just similar, just we prepare our samples and make these samples is amplified by PCR, and after that is a visualization. For PCR use electrophoresis, and for LAM using the real-time fluorescence. And in this case, I use this machine, Genie 3, and this is the primer. Maybe the difference of the concept of the PCR and LAM is in this method, that in PCR we just use the two kind of the forward and reverse primer, but in here we need 4 kind of until 6 the primer can conduct these samples. This is just the figure out the mechanism of these methods, and this is during the field survey. We collect samples with the symptom of Begomovirus, and directly on site on this field, we use the Genie 3, this machine, for detection of the samples, and we get the data that some of them has the positively infected. If we want to compare about the LAM and the PCR result, so we can know that the result is same. So this is become the effectively methods, I think, for LAM to apply it, become the rapid and simple detection assay under feed condition for routine sampling of plants. It means that our farmers in the field also can make these methods directly and get the result quickly. I think this is all of my presentation, Mr. Tian. Thank you very much.
Okay, thank you very much, Mbak Fariha, for the presentation. And we'll continue with the last presenter for this session, for Mrs. Eka Sulistiowati. I will invite Mrs. Eka Sulistiowati.
Hello, good morning, everyone.
Okay.
Can you hear my voice?
Yes,
Is it
very
clear?
Yes, very clear.
Okay, thank you. Thank you.
Okay. Mrs. Eka Sulistiowati currently is a lecturer and researcher at Biology Department, UIN Sunan Kalijaga, Yogyakarta, and the topic of her presentation today is about climate change and biodiversity. Mrs. Eka Sulistiowati, the time is yours, 25 minutes from now, please.
Thank you very much for the opportunity. Let me allow my share screen. Is it sound yet? Can you all see my screen? Moderator,
yes.
can you help me?
Yes, it already appeared on the screen.
Okay, so my presentation is titled Dangerous Decline, Climate Change and Biodiversity Loss, taking an example or perhaps case study in Indonesia. And before I go to my presentation, I would like to introduce myself. I'm Eka Sulistiawati from Department Biology, UIN Sunan Kalijaga. And my field of research is actually environmental sciences, especially environmental management and biodiversity conservation. Before I start my presentation, I will give you a little bit about what I'm going to talk about today. First of all, I will talk about climate change, maybe just a little bit because already a lot has been known about climate change, especially in this rapid era of research dissemination. And the second one, I will also talk about human-induced climate change and how it affects biodiversity loss, how it led to biodiversity loss in Indonesia. And the second one, what actually the mitigation and risk management plan has been put on site in Indonesia, especially leading to the community resilience. But before I go to my presentation, I would like also to show you a video about Indonesia and the situation right now. I hope you can enjoy the video before I go to my next slide.
Indonesia is not just wonderful. Indonesia is a wonderland.
Well, that's the video that I prepared for you all. I hope you can enjoy it. And I will continue to my slide. Wait a minute. Slide. As I said before, I will talk about a glimpse to climate change first. In Indonesia, climate change is mostly, caused by fossil fuel, whether it's in transportation, electricity, and industry. It's also very common that we experience deforestation, although the rate is already decreasing right now, but it is a major effect or major cause of climate change in Indonesia. Because of that causes, we experience right now temperature rise, change in rainfall pattern. We also experience sea level rise. And basically we have climatic anomalies in Indonesia. In Java alone, today in September, we still experience rain, non-stop rain from July, August, September, which is quite uncommon because usually it is dry season, but we have a very, very wet dry season this year. And for some experts, it says that the climate indeed has changed. Because of that, we have the climate change induced disaster. We have tornadoes, more frequent tornadoes. We have more frequent fire, also landsliding, flooding, and etc. Which at least we have the problem in water, energy, and food scarcity. And the most alarming one, we have biodiversity loss. which in turn, in the future, it all is a threat to our sustainability, especially the sustainability of the planet. Let's take a look about the rainfall reduction in Indonesia. The color blue shows the percentage of rainfall reduction in Indonesia, and the yellow one show an increase. So basically, climate change has changed the pattern. Some area that generally more wetter than before, and some area it's kind of like drier than before. Because of that, some of the lakes in Indonesia may also be affected by the changes in rainfall-rich area, blue, Bengkulu, South Lampung, part of Java. It experiences 20% of rainfall reduction. An increase is experienced in south of Aceh, part of Sumatra, Kalimantan, and Papua. Several lakes, they are greatly affected by the changes of rainfall pattern and also the changes of precipitation. And it led to crisis in water reservoir in Indonesia. The data is projected by BMKG. In 2013 and 2039, there will be quite alarming decrease, minus 20% of water will be available in some waduk or water reservoir and situ, and also lake in Java alone, Java and West Nusa Tenggara alone. So the number in the future, the prediction is not really nice. It's kind of like gloomy and sadly. And we also experience the surface temperature rise in Java Sea, Flores Sea, Banda Sea. The trend is a bit increasing from 1986 to 2013. But what will happen actually to our biodiversity if we couple biodiversity and climate change? Biodiversity itself is the variability among living organisms, whether it's in terrestrial, in our marine ecosystem or aquatic ecosystem, and it includes diversity between species and the ecosystem. All of this diversity, if we couple them, if we add them one factor, which is climate change, what we will have is the impending disaster to biodiversity. I'm afraid that we actually already experienced what we call silent extinction. Some of the species which are not been in our which are not studied in our database, they already had gone because of the changes of climate. Even before we know them, even before we had developed the database of them. This silent extinction actually because of the changes or the chaotic system that occurs in the overall sphere, the overall ecosystem in our planet. Climate change, it affects atmosphere, which also affect cryosphere, affect hydrosphere, lithosphere, and biosphere. At least we have melting of glaciers in our cryosphere, we have sea level rise in our hydrosphere, we have flooding in our terrestrial ecosystem, we have forest fire in our terrestrial ecosystem, drought, which led to later food insecurity, biodiversity loss, water scarcity. So the impact is not only to our biodiversity actually, but its overall socio-economic development impact. But in terms of biodiversity, we know that biodiversity consists of genetic diversity, species ecosystem, species diversity, and ecosystem diversity. All of these are threatened by what we call as the disaster due to climate change. The system is quite chaotic right now. This is based on the IPCC report in 2019. First, in terms of ecosystem, my video perhaps showing you some ecosystem that I have highlighted, forests, sand dunes, ocean. But now I will talk about forest cover loss. Indonesia primary forest loss in 2002 and 2020 somewhat. It firstly increasing, but now thankfully the trend is decreasing. However, although it is kind of like happy, happy situation that, okay, it's already declining Indonesia. Primary forest loss is not really that bad right now, but it's still Indonesia is the top 10 countries for 2020 primary forest loss. The number we see. We in the fourth ranking of top 10 countries of primary loss. The data is from Resource Institute. So how? Why this happen? Yeah, because of deforestation. So many forests are, open for industry, for agriculture, for plantation, palm oil plantation, rubber plantation, maybe development, human development, buildings, etc. So the cause is our primary forest loss. I hope in the future this number is continuing to decline actually, because if we have a decline in primary forest loss, it means that our country will out of the top 10 ranking. And the second example I will give you is the mangrove forest change. Mangrove is really important in Indonesia because it provides food, it provides protection from flooding, it provides protections from tsunamis. It's also part of the cultural mechanism of our people. So, but in mangrove forest, the figure is not really exciting. It's not really comfortable to read. You see that the deforestation and forest degradation increasing over time. Yeah, because of, we have maybe opening of mangrove forest for, development, human development. I do not know whether it can be improved in the future, hopefully, because we have the missing data from 2008 and 2018 forward. So we hope that the data will be slightly comfortable later on in the future. And next, in terms of species, diversity of species, Indonesia is part of Asia Pacific. And the total number, which will be listed as endangered, critically endangered or vulnerable species in respectively 148, 300 to 739. This only the known species. What about the unknown species? Perhaps the number that will be listed in the IUCN list of endangered species will be much more, many more perhaps. And the media also highlighted several, uncomfortable, inconvenient issues like primate species may become extinct by 2050, long-tail macaca. It become a very shocking, painful, but predictable news. We all already predicted that, there will be extinction, but it's still painful to see that all of this beauty are gone in the near future. In terms of genetics, I highlight the example of medicinal plants. This is research from Ignatius et al. 2022, a relatively new species. Because of the population size is reducing, they lost 30 to 80% of the distribution area. More will be assessed as vulnerable and endangered in the future. You see, here, okay, sorry. The species in here, the Sumatra, this is the figure in 2030 and 2050, above the prediction in 2050. And in 2080, there will be less and less species will be observed in Indonesia, medicinal plants. So we lost a lot of genetic resources then for medicine purpose, for the material for building medicinal drugs or that kind of thing. In Papua, see, more and more species will be categorized as vulnerable or endangered. But what all of these have related to do with agriculture then, as the topic of this, symposium today? In agriculture, first, climate change, surely it increased flooding, it rise the sea level, increased drought, it increased desertification, increasing temperature, carbon dioxide, and etc. And the consequence is we have an increased disease severity. Perhaps the plant virus, which highlighted by the second presenter, maybe also lowering of the yields of our crops, the lower nutritive quality of our food. That's the consequences of climate change. In a longer term, IPCC in 2014 made a projection or made an estimation that in a longer term, there will be decrease in yield. This is yield. Decrease in yield, and this is an increase in yield. You see that today we are here. The increase in yield is relatively equal to decrease in yield. But over time, decrease in yield will be much more overshadowing the increase of yield. So the situation is not really good. Today, even we already experience food crisis in some part of the world, but in the future, the prediction is quite negative. It's not really a good number to read for the projection in the future. It's only, maybe 80 years from now, not far away, maybe 80 years from now, we just losing our yield. And in relation to insect, which perhaps already highlighted by the first presenter, because of carbon dioxide increase in our environment, surely we have an altered C and N ratio, carbon and nitrogen ratio, which means our crop today has a low nutritional quality foliage. For insect, it's mean for them, they have greater consumption rates. If they consume a very low nutritional quality foliage, then in the longer term, they will increase in development time, metamorphose perhaps, which generally only takes 3 months, probably it takes longer than that. Then the result is we have a decrease in pest abundance. We have a decrease in insect abundance, beneficial insect, perhaps the pollinator, we have a decrease in the future. And the second one, the changes in precipitation creates heavy precipitation. We have only either too heavy or drought. If it's heavy, then the insect washed away. If it's heavy, then we have a decrease overwintering. But if it's drought, plants more susceptible to insect attack. Perhaps it's also more susceptible to virus infection. It's an area needs to be researched further. What are the consequences? What are the impacts of climate change to insect itself, to the prevalence of virus attack in plants? That's the future research perhaps. And in terms of mitigation, risk mitigation, and also mitigation plan, risk management, we actually already have the international, the global framework, the global consensus to tackle climate change issue. First, in UNFCCC, we have the framework for climate change adaptation. And in terms of 2030 agenda, we already developed sustainable development goal. Sendai framework help us to, seeing the disaster risk reduction due to climate change. All of them with the one goal, reducing vulnerability, and also enhancing the resilience of community, especially local community against climate change. In the national level, we already first in 1992. Excuse me, can the there's noise in the background. Okay, thank you very much. In 1992, we already have Rio Earth Summit, which has resulted in the United Nations Framework Convention on Climate Change, UNFCCC, established in 1992 in Rio de Janeiro. And Indonesia has already ratified this consensus. Wait a minute. This consensus in 2004. Even so, we already in 1997, we already have Kyoto Protocol and adopting Kyoto Protocol. And in 2015, where the Paris Agreement occurred, we ratify Paris Agreement in 2016 by Act number 16, 2016. And we also developed the first nationally determined contribution to UNFCCC. And we recently submitted our updated NDC to the UNFCCC. It means that we already have the global and also national commitment to tackle the issue of climate change, to help the nation build resilience to climate change, and also to build, mitigation against climate change. But what actually the community or the local community do in regards to climate change itself? First of all, they develop strategies actually. Strategies which already long been embedded in the practices of daily people, especially in Indonesia. This is my research, part of my research. And I would like to highlight now. The strategy number one is agroforestry. Several communities, especially in Java, including in Sumatra, Kalimantan, and also in East Nusa Tenggara, they developed what so-called traditional agroforestry system, or TAS. In TAS, my case study for my research is in Gunung Kidul. Agroforestry in Gunung Kidul is improving over time. I analyzed data from 2002 to 2019. The area planted for agroforestry in Gunung Kidul is quite increasing. And it has been, proven that the agroforestry could maintain the biodiversity environment, biodiverse environment, because we have so many plants and animals which can be protected by agroforestry. By that, we also ensure the sustainability of livelihood of local people. And we have so many examples of flora fauna that can be protected by agroforestry. For example, this is burung kutilang and burung cendet in bahasa Indonesia, or in a Latin nomenclature, it's called Laniosa. And we have, several commodities that can support the livelihoods of the people, especially the high valued teak or jati. We also the highly valued cacao or cashew, and we also have cocoa, the resource for making chocolate, the Indonesian chocolate. And it's really, it's really a traditional strategy, but it helps people, especially local farmers, to adapt and to, mitigate the effect of climate change itself. And we also have pertanian cerdas iklim, or climate smart agriculture in Indonesia, every part which is initiated by the Ministry of Agriculture. Basically, pertanian cerdas iklim or climate smart agriculture, CEA, is a practices that benefit food production. The basic framework, it tries to increase food production. At the same time, it adapts to the climate change, and it provides mitigation to the effect of climate change itself. The practice, for example, in adaptation, we have diversification of crops, livestock, fisheries in one location. It's usually an integrated farm. also we have improved on-farm and off-farm food storage, lumbung pangan, we call it in Indonesia. We have also expansion of agricultural land, but sustainable expansion of agricultural land. We increase the use of mechanization, fertilizer, and other inputs in an optimal way. And in mitigation, the CEA aims at reforestation, and we have, kind of like producing woods and etc.
Excuse me, Bu Eka, 2 minutes more for the presentation.
Okay, I just only have 1 or 2 slides. Okay, so we have increased productivity, enhanced resilience, and reduced emission. That's the the strategy for CAA, the final goal for CAA. I will just skip this slide. And we also have local strategies to tackle climate change by having local people practicing their local knowledge in farming and also food production. Campaigning for local food, campaigning for local species for their food sources, also campaigning for local practices of traditional farming and acknowledging them, incorporating them to the climate smart agriculture is one way and one strategy to tackle the effect of climate change. It's a very sustainable way to protect our environment. And the last one, okay, good. We have already come to our conclusion. We know that climate change is predictable, but the biodiversity loss as a consequence of it is inevitable. It threatens the sustainability of the planet, and certainly we need mitigation and risk management of the consequence that climate change has posed us into. The last one, there is always plan B, but there is no planet B. We only have one planet. The Earth is your only plan. That's it. Thank you very much for the opportunity to talk to you all. Please welcome if you have any question or discussion. Thank you very much, Ms. Dian. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Bu Eka, for the presentation and for the very remarkable closing statement. Okay, and we will continue to the session for discussion. And in the chat box, we have several questions. I will read the questions for you. The first question is from Ibu Kadarwati. I think this question is for Mbak Fariha. Okay, the first is, this species under study, Begomovirus, a species with ornamental plant virus begonia. And the second, what factors and how to solve them? And the third, in what climate does the virus usually attack? Maybe Mbak Fariha, you can answer this question directly, please.
Okay, thank you for the question, Ibu Kadarwati. This is my director in Instiper, and Mbak Dian Aruni, that has already. So I get the first question and the one only of the question. Thank you. So for this question, this is a good question. How about the correlated with the begonia plant of the ornament? So in the world, the kind of variety of the ornamental plant is reported infected by the begomovirus. But unfortunately for the begonia, I have already recorded. And in Indonesia especially, some of the reported of the begomovirus infection is focusing on the crops, like tomato, chili, melon, beets, and something like that, and not yet, I think, not yet focusing on the ornamental plant. So this is the big big problem for us, and to solve this one. And the next question is, how about to conduct this virus? So there are many kind of reported for describe the mechanism of the infection of the begomovirus. Some of them describe that the genetic modified, I means that the like CRISPR or something like that can be used to eliminate the gene that infect for the pathogenesis of this virus. But the other ways is can be also conducted by the management of the insect spreading. So maybe Arisa also study about the insect because the one and only possibility of the spreading of these viruses is come from the whitefly of the insect vector. So the most important one is how to control this insect. But unfortunately, also in Indonesia, we know well our farmer usually has the huge of the land in the open area and no mechanism for like just little one like use the trapping or something like that. So this is become the I think the good point if the farmer can control of this insect. Some of research also conducted use the like barrier plants that plant surrounding in this land, and we can make sure that this barrier plant is not become the host ring of this become virus. So this is become the easily methods I think that can be conducted for our farmers. And what the season in our country is very affected by this virus, especially for a bigomo virus, because this is very correlated with the transmitted of the whitefly vector. So the summer season, this is the best condition for whitefly to breeding and spread rapidly. So automatically, this is will be effect of the spreading of these viruses and become more severe and severe, like that. Mbak Dian Aruni, thank you.
Okay, thank you very much, Mbak Fariha, for the answer. And the next questions, there are some questions for Dr. Arai, but he already answered in the chat box. But if you want to, would you please elaborate more about the answer from the first question? About the controlling chikungunya and also the other question. Okay, please, Dr. Arai.
Yes.
Kan juga ikuti palan tempi pas buka pertanyaan toh.
Sorry. So regarding the question, maybe the chikungunya virus is belonging to the fast run DNA virus, maybe in the first run, so similar to the dengue virus, so might be possible to control. And similarly, the dengue and Zika and such kind of virus are mainly the first run single strand DNA viruses, but it is quite difficult to control the double strand DNA viruses actually. So in my institute here, there is some injection machine behind me. So some people are just injecting Obakir in the several pest insects to control the plant viruses. Maybe Fariha-san is doing the similar Begnovirus, yeah?
Yes, I asked.
Yeah. So maybe some similar attempts want to be conducted in Japan also, but it is a bit hard for depending on the species of the insect. For instance, the Lepidoptera is very hard to maintain Obakir in the insect. However, Hymenoptera stinkbugs is very easy to inject and release in the wild. But because Japanese government is prohibited to release such kind of things, but I do not know if such kind of law is still existing in Indonesia. So that is my interest. And the question is to detect, yeah.
I think the second question is how we differentiate Wolbachia infection into different insects. Yes.
If I do not know if my answer is correct, but so might be distinguish the Wolbachia strain by using PCR and sequencing technologies. But if we want to infect the Wolbachia into the different insects that I maybe showed before, that to inject the Wolbachia into the by using the microcapillary and to the other insect species. So. And the third question regarding the Lela-san,
Yes, the
asked me
CI.
is very complicated. Actually, this is quite a difficult question because the CI mechanism was only revealed in the Obakir, but the gene, the causative gene of the CI inducing Obakir had not been identified from other bacteria species showing the CI phenotype. So the mechanism is actually completely different from Obakir and the other bacteria. And similarly, I found the male-king gene of the Obakir, but that was not found from other male-king endosymbionts. So maybe the bacteria or other kind of viruses have developed the similar but completely different system to manipulate the insect species. A similar phenotype could be, I think I might read the similar paper that's showing that the plant might, the contrary of the plants is very large, and that have a conflict between the genetic factors of the host plant. So similar phenotype of the CI could be found in the plants. And recently, a researcher in France reported similar phenotype in snails. So this is my knowledge.
Okay, thank you very much, Dr. Are, for the answer. Is there any more question? We still have 1 minute. I think that's all the question.
So can I ask a question?
Okay, please.
For Hariha-san, and you showed the virus infection in the plants, and I want to know if the Begnovirus have a host specificity, and if so, how about the phylogeny? For instance, Begnovirus found from the tomato or Begnovirus found from other host species is similar in the phylogeny.
Okay, thank you, Alexander, for the question. This is this is I think the best moment for discuss together between the phytopathologist and from the insectologist and entomologist. This is the interesting question. This is right, Sensei, that the Begnovirus is very small genome, and it is also very easily to mutate or maybe recombinant with another type, another isolate, something like that. So for example, like the firstly reported from tomato, however, increasing by the number of the member of the virus, this is also become in fact the another of the family of this plant, the similar one. But if we check detail about the molecular genetic, about the especially for DNA, that can vary in fact for the pathogenicity of this virus. This is any kind of the differences of the gene. So maybe the next project is about the SNAP analysis and CRISPR method for now about how about the diversity, the member of the Begnoviruses from the like similar of the host ring or maybe the another of the host ring, something like that. Araisan.
Yeah, thank you.
Thank you.
Okay, thank you. And this is there's last question for Bu Eka. In general, on environment in Indonesia, in various ecosystems, forests, freshwater, sea has decreased in quality both by climate change factors and anthropogenic factors. What strategies and indicators do we need to ensure the sustainable use of these ecosystems? Please, Bu Eka.
Thank you very much for the question, Bapak Nasaruddin from FMIPA UHA. The answer is quite, difficult because we have so many strategies and indicators if we are talking about the, environment as a whole, because we have so many ecosystem, forest, freshwater, sea, and everything perhaps has its own strategy. But I will kind of like respond the question from a general or general terms. In ecosystem, we know the term which has been proposed recently in new conservation era, which called ecosystem services, which mean that each of our of our ecosystem provide us with tangible or
Bu Eka, are you still here? Okay, I think she lost her connection. And Bu Eka, okay, maybe we can continue the answer in the chat box later, because we already come to the end of the first session, and from this session we can conclude that there are so many methods that can be used to enhance the agricultural and environment resources, and also it is important for us to apply mitigation and risk management in this era of climate change. Okay, thank you for all of the speakers for this session. And before we end this session, I would like to
I think I'm just losing my connection.
Okay, that's okay, Bu Eka, because we have a limit of the time, so maybe you can continue the answer in chat box later.
Okay. Sorry.
Okay, that's okay. Thank you. And before we close this session, the first session, I would like to give the certificate for the appreciation for the speakers, represented by the certificate for Dr. Arai for the committee. Please share the certificate. Okay, thank you very much for Dr. Hiroshi Arai and all of the speakers for the valuable talks today. And I think we already arrived at the end of this session. Thank you very much. And I will give the screen back to the Master of Ceremony. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much, Ibu Dian. For next agenda, we will have short break, 3 minutes breaks. Okay. Ladies and gentlemen, we are now continue to the next panel session. This session will be moderated by Ibu Lela Susilawati. Bu Lela, please, time is yours.
Ya, terima kasih, Raisa. Okay, good afternoon, all of the participants. Kayaknya suara saya. Good afternoon, all the participants. Thank you very much for still joining in this Zoom on mini symposium 22 on sustainable agriculture and environment. So now we move to the second panel session. In this second panel session, we have three speakers as session one. So we have here Dr. Dian Armanda, then Dr. Eric Firmansyah and Alice Zah from Russia. But I still couldn't see the speakers from Russia here because we here in Indonesia 4 hours ahead of Moscow or Russia. So maybe later on Miss Alice will join because now in Russia still morning. Okay, before we start, let me introduce myself. So my name is Lela, so I will be moderating on this session. And please to all of the audience to mute and also we will have designed time to address questions, but you may submit questions earlier by maybe, after the speakers finish on their speak, and you can write down your speak on the chat and I will read it for you. And in Indonesia it's also fine, so I will translate it for you. If how to say, but I think maybe English is fine for all of the audience here, right? Okay. Then for the first speakers, I will welcome Ibu Dian Armanda, already here, right? Okay,
yes, I'm here.
Terima kasih Bu Dian. Waalaikumsalam warahmatullahi wabarakatuh. And the second speakers, Dr. Eric. Okay, Pak Eric. Selamat siang.
Selamat siang. Does my voice heard clearly?
Yes, very clear, very clear.
Okay, thank you.
Okay, and the third speakers, I don't think that Alice is already is here. Yes, I said now in Moscow still very early morning, so maybe after that. Okay, for now, so I will give the chance for the Ibu Dian to deliver your speak in 25 minutes. Please, Bu Dian, time is yours.
Thank you. Can you see my slide now?
Yes, clear.
Thank you, Ibu Lela. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, all speakers and participants. Thank you for the opportunity to present a part of my research here. To start this presentation, I would like to start with some questions maybe for you. What did you eat for breakfast this morning? Let's say fried rice. It could consist of some ingredients such as rice, onion, maybe garlic, chili, a little bit of coconut oil, maybe some chips and some lettuce leaves as toppings and extra vitamins. Have you ever wondered where those each ingredients come from? From which farm or which city? If it was, of course, distributed from other city, or which country if it was imported, and what method did they use to produce it? Have you ever wondered what and how much water and maybe material, fertilizers were used to produce that food? Maybe it's quite intriguing to answer the question. In this In this presentation, I'm going to explore the recent innovation of farming technologies worldwide and how the life cycle of food deserves more of our attention. It is regarding how it is produced, processed, distributed, consumed, or even discarded or recycled. So we can see a whole life of a food product from a bigger picture, particularly if it is produced in and around cities, not in rural area or village, using the emerging growing practices such as hydroponic, aeroponic, and etc. Therefore, I title my presentation as Exploring Innovative Sustainable Urban Agriculture, Trends and Challenge. The main source of this presentation will be particularly taken from this article, our systematic review article published in 2019 in the Journal of Global Food Security. Okay, let's first clarify this terminology before we proceed. Urban agriculture, what is it? Urban agriculture is known as the production, process, and distribution of food and other products by plants and or livestock, not only plants, of course, and or livestock too, raised in and around cities to meet local needs. So it means urban farming are solely intended for meeting local demand only, not for not to be exported and etc. Urban farming itself has a very long history. If you are interested about this history, you can find a historical overview of urban agriculture in the supplementary material of my review article. The transformation from rural to urban agriculture began around 2010, and will be dominated by specialized small and medium urban like hobbyist farms and the emergence of large or commercial agriculture. Urban agriculture is expected to be the typical type of agriculture in the future. In 2020, researchers also found that COVID-19 outbreak in early 2020 has supported the emergence of urban farming practice in cities worldwide. The combined effect of cities lockdown, movement restriction, increased food losses, and export costs foster cities around the world, especially the non-self-sufficient countries, of course, to maximize their local production, especially for perishable foods such as vegetables and fruits. Despite the needs of producing more and more food for the more and more urban needs in the future, urban agriculture practices are still facing pro and contra. Are you pro or contra? Some researchers shown that urban agriculture has a contribution to food security and food resilience. It reduces food miles and reduces economic pressure. It creates jobs. It supports education, it supports community health, women empowerment, urban beautification. And nevertheless, urban agricultures are still very challenging too, because regarding of some aspects like the competitive land and water use in urban area, pollution risk from the urban ecosystem to agriculture and the other way around, contamination of food products by heavy metals and organic chemicals, and increasing health issues due to sanitation and vector disease. Some farmers are trying to solve these challenges by applying novel technologies, which then we knew them as innovative urban agriculture technologies. So here we define innovative urban agriculture itself as urban agriculture that optimizes food production by minimizing maintenance and resource whilst maximizing yield by involving at least one of the recent technological innovations in open or closed system. Here we can see the types of urban agriculture. Urban agriculture comprises many types. It can be conducted with or without farming control to control the environment like light, maybe fertilizers, media, and etc. Then sometimes greenhouses are commonly used for controlled environment agriculture. In the most advanced agriculture, we might see the integration of sensors and the internet to gain precision in farms. Based on area usage, it can be conducted in any indoor space, such as abandoned building, abandoned bunker, parking lot, basement, and etc. Or outdoor space, such as backyard, front yard, balcony, rooftop, and etc. Vertical space, like staircase, outdoor, and indoor walls. Urban farming also can be integrated with the building itself. It utilizes rainwater catchment for watering system. It uses electricity from solar panel system. It uses gases recirculation from the office area of the building to and from the farming area, and etc. So there are so many techniques can be used in urban agriculture. For instance, soil-based farming, soilless farming, such as hydroponic, aeroponic, aquaponic, or mixed between them. Yeah, some questions urban challenge are well answered by innovation in urban agriculture. We mentioned before about the competitive water use. This is solved by hydroponic. Researchers claims that 90% less water than the field farming is gained when we use hydroponic. Or aquaponic, when we integrate fish and vegetable farming. Or aeroponic, which use mist, water mist, or like nutritious mist for the plants. Pollution risks from and to the other urban ecosystem can be solved by closed system or making the agriculture of the soil. So we do not use soil at all. Increasing health issues due to sanitation and vectorial disease also can be solved by this kind of system, by using the closed system. Like we see here, there are some techniques we use here: terraponic, hydroponic, aquaponic, aeroponic. All of this is categorized as closed system. Most urban agriculture choose horticultural products such as vegetables, herbs, and small fruits because they are perishable, has high economic value, relatively lightweight. So this is a very proper to be hanged in the hydroponic rack. Then short season, simple and practical to grow. And next, there are still some challenges that can be solved, like competitive land use can be solved by vertical farming, rooftop farming, building integrated agriculture, like I said before, indoor farming. Then contamination of food products by heavy metals and organic chemicals also can be solved by precision agriculture, because with precision agriculture, we can optimize the use of fertilizer itself. And thanks to these technologies, which provide solutions not only for these challenges, but also to optimize the productivity itself, the desired quantity and quality of the crops or the produce itself. Internet of Things and sensors enable to ensure that crops and soil receive exactly what they need for optimum health and productivity. Many growers are using fertilizer recipe to yield produce with certain flavor. Maybe you like, maybe we like our lettuce to be like sweeter, a little bit sweeter, or a little bit sour, or crispier, or even greener. We can we can follow certain fertilizer recipe and light recipe too. Yeah, researcher and many lab companies are developing light recipe, which enable us to regulate the growth of our crops or plants. Yeah, light recipe is a certain recipe of light. Sorry, I didn't. I don't have any intention to like promote one of the brand here. So Philips is one of the industries that develop this kind of light recipe. Light recipe is certain recipe of light, spectrum, uniformity, and duration of exposure itself or time. Yeah, in to grow crop in desirable way. If we need to produce more leafy greens such as lettuce, we use more blue light than red light. If we need to produce more fruit like tomato, we use more red, maybe in combination with the blue one, yeah, creating violet light, yeah, to grow tomato. So here we see that blue light mostly promoting leaves growth or leaves growth or for vegetative growth, and red light mostly promoting flowering and fruiting or generative growth. Okay, commercial scale of innovative urban agriculture is the most challenging scale to be studied, of course, commercial scale. In 2019, we had tried to summarize production information from 18 innovative urban agriculture practitioners worldwide. We studied more than 100 sources, mainly from scientific articles, books, and more than 40 news media and websites to conduct this review. Some also from private conversation or private conversation via email to the grower. There are some interesting findings from this data. Firstly, all of the 18 practitioners apparently apply indoor vertical farming. 9 from the US, then 4 from Europe, and 5 from Asia. And I'm sorry, no available information from Australia and Africa. Yeah. 9 practitioners are from the US, and altogether is operate at least 28 farms throughout the country. Yeah. Secondly, although some practitioners started their research and development before 2010, most of them started the commercialization around 2010. But apparently, 3 of them were no longer operated since around 2017 and 2018. 2 farms closed because of their financial problem, and 1 farm, the one in Japan, closed because of the company moved on to address the next issue, focusing on the dairy industry. So they closed down their industry. Thirdly, the highest actual food productivity is estimated to be achieved by aerofarms. In New Jersey, the US, which apply aeroponics and conducted fully underground in the basement. Their production capacity is up to almost 140 kilograms of vegetables per meter square per year. This is a very big amount of veggies. Fourthly, mainly produce leafy vegetables, microgreens, followed by herbs, fruits, mushroom for local market, then grocery stores, restaurant, hotels, and etc. Okay, this is the table that shows the technology used by these 18 practitioners. All of them are indoor and vertical farms. In the left are aerofarms, which are using aeroponic to produce almost 140 kilograms of leafy vegetables per meter square per year. This is the most productive farm so far. This farm is fully operated in the basement underground This data shows us the high potential of aeroponic than the other technique. The second most productive farms is 80 acres farm in the US, which applied some indoor vertical hydroponic farm to produce 81 kilograms per meter square per year microgreens. Yeah, apart from this data, in Indonesia, we already have actually agri-farm, which have been developing precision hydroponic in greenhouse since early the year 2000. They can produce up to 60 kilograms of cherry tomato per meter square per year. And this data was never been published anywhere else before because I gathered the data by doing direct interview with the owner. This is shown that there are still many more farms outside that have never been published widely in the internet. Now we have a question. Are these innovations for agriculture sustainable or not for our planet? Yeah, current agricultural activities together with forestry contributes to 24% of our greenhouse gases emission that cause global warming. This is only seen from the greenhouse GHG, greenhouse gas emission or climate change perspective only. In fact, there are more ways than CO2, not only CO2. There are more impacts in environment and an ecosystem that rarely mentioned in many publications. For instance, biotic and abiotic depletion, acidification, eutrophications, photochemical oxidations, then ecotoxicity, human toxicity, cumulative energy demand, and many more. Yeah. From the surface, maybe we see innovative urban agriculture farms seems to use less water and soil than regular farm. Moreover, a study revealed that like drainage water reuse for greenhouse hydroponic cucumber production has increased water efficiency up to 33% and reuse blah 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 kilograms and blah 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 kilograms and blah blah kilograms of fertilizers at the farm. But if we see it from the bigger picture, from upstream to downstream, they may require more water, material, and energy than conventional urban agriculture to properly function as a complete food production system. They may not as greener as it seems. For example, a study revealed that an urban hydroponic system required 82 times more energy per acre to produce 11 times more lettuce than regular agriculture. Therefore, if a farm needs to produce lettuce because of the high demand of lettuce in the market, of course, the environmental burden of the production of 1 kilogram lettuce should be assessed from its whole life cycle systematically. We cannot separate each stage of the production. This can be done with lifecycle assessment method. Here, I introduced the simplified lifecycle of a product consists of some stages. Like LCA, we can optimize its stages to have a greener agriculture, from extraction of raw material, then go to manufacturing, then packaging and distribution, product use, and of life, then back to the environment and back to the extraction of the raw material. Yeah, this is the life cycle thinking that we should concern. Life cycle assessment is a well-defined method to calculate the environmental impact associated with all the stages of a product. The principle and framework of this method have been standardized in ISO 14040 and 14044, since 2006. We can use LCA to do environment productivity trades-off. Yeah, so we do a trades-off between environment and productivity, whether the technology is worth it or not to be applied. The steps are defining goal and scope of the assessment, then doing inventory analysis, defining its impact assessment. We can choose some impact categories to be applied, such as maybe greenhouse gas emission. This is the most popular one. Yeah. Then biotic and abiotic depletion, or maybe acidification, eutrophication, or ecotoxicity, and etc. Yeah. Lastly, we go to the last step, that is the interpretation step. After finishing all steps, we can use the results of the LCA to plan a greener system, not only for the production of food itself in farm, but can also for the distribution, marketing, and other stages. Yeah, nowadays, LCA has developed as an important aspect in LCSA, Lifecycle Sustainability Assessment, which comprise life cycle assessment, of course, environmental life cycle assessment, and life cycle costing, which is seen from economic aspect, and social life cycle assessment, which is seen from social aspect. This is quite complicated to be done. Yeah. Every farming system requires agricultural input, such as water, media, fertilizer, maybe pesticides. However, innovative urban agriculture, which typically is a closed or semi-closed system, often requires more sophisticated construction. Yeah, solid substrate as soil substitution. So we use like media like perlites or rock wool and etc. Yeah, equipment and automation. Yeah. For sensors and in the internet, yeah, all of this imply the use of more material and energy, of course, resulting in more activities and a more complex life cycle.
Dian,
yeah,
sorry to interrupt. You still 4 minutes left.
Yeah, thank you.
Okay.
For example, if a farm produces lettuce with a hydroponic system, the environmental impact of the production of 1 kilogram lettuce should be assessed from its whole life cycle. We should gather and calculate any inventory data. Inventory data consists of any what and how much input used and input used, yeah, and output produced, yeah, like waste, yeah, not only input such as water, electricity, fertilizer, seeds, maybe, but also the metal and plastic used for the construction, yeah. From the upstream process like extraction and transport of raw material, then we go to manufacture or of infrastructure itself, then transporting to the farm downstream, yeah, then we get into the farm, and in the farm after that there are also we also should think about any environmental effect like emission to water, air, and soil. So this is just the big framework, and I will show a small example. This data is an example of LCA of some relatively very simple, innovative urban agriculture system. This data was obtained from the environmental impact analysis, yeah, or SEA, of the production of 1 kilograms of tomatoes. So as it turns out, the more sophisticated the technology used and the more we plant away from the ground or soil, then it relatively increased the environmental impact as well. To produce 1 kilograms of tomatoes, we emit 2.2 kilograms of CO2 gas into the atmosphere. Can you imagine it? The amount of CO2 is much more than the produce itself. In comparison to tomatoes in the soil with compost as fertilizer, we save up to 3.26 kilograms of CO2 to the soil. We return this CO2 as carbon stock in the soil. Yeah. So here we gain something, yeah, because yeah, yay, we do it greener. We do it with compost and with the soil. But how long we should do this, yeah, using conventional method, because we should move to the next technology too, and to increase our productivity too, we should think about adaptation. We should adapt the new technology to make more food and make more, yeah, quality, of course, the good quality food for us. So I think this is the last sample I have, and it's gonna be the last slide I have. OECD ever mentioned in 2013 that urban agriculture is expected to be the typical type of agriculture in the future. So as a conclusion of this presentation, there are more questions, more room to explore about this innovative urban agriculture. We should do more studies on this area. LCA or LCSA, yeah, is very complex, of commercial innovative urban agriculture system. It could be various types, yeah, hydroponic, aquaponic, and etc. Yeah. Or the food product itself, yeah. For example, doing LCA to compare the imported rice and Indonesian rice, yeah, imported versus Indonesian rice, or maybe organic versus conventional tomato, like that. Our Ministry of Environment in early 2021 last year, has issued new LCA regulation for industry, including for food agriculture too. So there are many support from the government to do this. And sensor and IoT, of course, this is a very challenging topic, yeah, to be explored more. Precision agriculture for precision fertilizer and light, growing stages, flavor, and etc. Then we can also do economic sustainability assessment from using the life cycle costing method, or maybe if you like social aspect, you can also use social life cycle assessment of IUA. For example, now a commercial IUA tends to expand to peri-urban area, not in the central urban area. It expands to peri-urban and rural area. So we can do some research, the readiness of farmers to adapt to this IoT era, maybe, yeah, it could be very challenging to do. And for biologists, maybe, because I know most of you are biologists, yeah. Maybe soilless system is very something challenging, something new, yeah. Like plant microbes interaction, maybe, or for more spectrum, we can see it from the bigger spectrum, more study on the conservation of Indonesian wild type crops, along with these high yielded varieties, or HYV, yeah, high yielded varieties era, yeah, and many more collaborative multidisciplinary studies to explore regarding this topic. Okay, that's all. This is the end of my presentation. I hope this information is useful and inspiring for all of you. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much, Bu Dian Armanda, for your excellent presentation. But I'm sorry, we have to end of your presentation. Okay, then we move to the second speaker. So here, Pak Eric Firmansyah. Would you like to present yourself by yourself, or?
I will present by myself.
Okay, right.
Lila.
Oh yeah, please. Okay, time is yours, Pak Eric. Silahkan.
Okay, thank you. Okay, does my voice and presentation heard clearly?
Yeah, yes, very clear. Yeah, we can see it.
Okay, good morning everybody. Assalamualaikum warahmatullahi wabarakatuh. Thank you for giving me the opportunity to share in this symposium. An honor to meet you here in mini symposium 2022 on sustainable agriculture and environment. I thank the organizer for giving me the opportunity. Okay, first let me introduce myself. My name Erik Firmansyah. I am from Faculty of Agriculture in Stiper in Yogyakarta. I'm also work in Center of Palm Oil Science or PSKS, also in Stiper. In this presentation, I will share and discuss about the opportunity, challenge, and what we have tried to do to achieving sustainability in smallholder palm oil plantation, or in bahasa we call kelapa sawit rakyat. So mainly I will talk about smallholder palm oil plantation in the context of sustainability. Okay, first let's look at the profile of oil palm plantation in Indonesia. In 2019, oil palm plantation were dominated by large private-owned plantation or perkebunan besar swasta, which accounted for about 50% of the total land area, followed by smallholder plantation at 41% and state-owned plantation only at 6%. However, interestingly, the projection for 2030, there is significant change in the proportion of land tenure, where smallholders plantation take over the dominance of large private-owned plantation and rank first with a share approximately 60%, followed by large private-owned plantation by about 39% and state-owned plantation, which are getting smaller by 3%. Thus, by 2030, we can say that smallholder plantation will become a major player in the palm oil industry in Indonesia. So, if we seek to create a more sustainable management of oil palm plantation, then our main target is to concentrate it to smallholder plantation. This doesn't mean that private large plantation are excluded, but they have the resource, they have capital to achieve sustainability target, unlike smallholder plantation. Okay, this is productive oil palm plantation by typology in Indonesia. So private owned have 7.25 million hectare, or producing 30.72 million ton of CPO or crude palm oil. They share about 61.82% total national production, and smallholders followed in 4.83 million hectare, or about 16.75 million ton CPO. They share 33.71%, and last is state-owned plantation. They have 0.51 million hectare, producing CPO about 2.22 million ton. And the development of the palm oil plantation sector in Indonesia is an integral part of Indonesia national development. Continuous efforts are needed to optimize the productivity, profitability, as well as sustainability of the oil palm plantation, especially in smallholder. So the government in Indonesia already commitment to realizing sustainable management of oil palm plantation in Indonesia. In 2020, President Joko Widodo issued a presidential regulation number 44 concerning the Indonesian Sustainable Oil Palm Plantation Certification System. Then this presidential regulation further realized in the form of Ispo, or Indonesian Sustainable Palm Oil, as a certification system for the management of oil palm plantation that are economically viable, socially viable, and environmentally friendly, based on the prevailing law and regulation in Indonesia. So the government has basically tried to make this oil palm plantation managed with more attention to sustainability aspect. This presidential regulation also result of consumer pressure, of course, especially those from European Union and United Kingdom. Okay, if we take a look to the Ispo certification mechanism, smallholder are required to fulfill 5 principles and 30 indicators. Number 1 is compliance with law and regulation. There are 5 criteria and 7 indicators. Then implementation of GAP, or good agricultural practices. There are 11 criteria and 17 indicators have to fulfill by the smallholders. Then management of the environment, natural resources, and biodiversity. There are 2 criteria and 3 indicators to fulfill. Then implementing transparency, 2 criteria and 5 indicators. And lastly is sustainable business improvement. There are 1 criteria and 1 indicator. Nah, this is Ispo performance by March 2021, issued by BPDB Kelapa Sawit. There are in private plantation or private owned plantation, 5.45 million hectare out of 8.68 million hectare already Ispo certificated. In estate plantation, there are 0.32 million out of 0.98 million hectare already have Ispo certification. But in smallholders plantation, only 0.19% out of 6.72 million hectare that already Ispo certification. So this is very small fraction of smallholder that already Ispo certificated. And Ispo is mandatory for all type of oil palm plantation, regardless there is a smallholder, estate plantation, or private owned plantation. Every typology of palm oil have to Ispo certification in 2025. Now, so by 2025, all smallholder oil palm plantation in Indonesia must be Ispo certified. Today is 2022. Only have about 2.5 years further for smallholder to fulfill this requirement, but there are only 0.90% that already Ispo certified. So the question is, what happens if most smallholder oil palm plantation fail to meet the deadline? Because the reality is that independent smallholders are still not ready and face many obstacles in fulfilling the assessment aspect in Ispo. Compulsory implementation of Ispo, I think, without improvement and assistance to smallholder, will have the potential to exclude or to discriminate smallholder from Indonesian palm oil supply chain. And I think this will be a disaster for oil palm farmer in Indonesia, because they will have a lower price in fresh fruit bunch pricing. Therefore, it become a challenge for us to help the government and oil palm smallholder also to quickly fulfill the assessment aspect of Ispo certification in 2025. Now, before we move on to discussing what thing might be done to support oil palm smallholder, we must first understand the reason why farmer in Indonesia choose this commodity, this palm oil commodity, over other commodity that already there. Number one, we have to understand that palm oil is a very quick, rich, productive age. In 29 to 28 months after planting, palm oil already already come to production stage. So farmer can harvest their plant in 26 or 28 months after they are planting the seedling. And number two, productive age of palm oil reach more than 20 years. In some cases, this palm oil can produce until 30 years before they have to replanting. Number three, this plant is very adaptive to various type of soil and climate. So the palm oil can be planting in sandy soil, can be planting in peat soil, can be planting in the hilly topography, can be planting in mineral soil also, and there are various type of soil palm oil still can productive. And number four, miraculously, this type of plant have few major pests and disease, not like other crop in Indonesia, especially. This palm oil have few major pests and disease. So there is not no pest or disease that can destroy 100% palm oil crop. Yep, maybe some pest or disease can destroy maybe about 30 or 40% maximum, but there is no pest and disease that can destroy 100% of the crop. Not like other crop that can be destroyed 100% by pest or disease. This palm oil is not. And the last, why farmer choose palm oil commodity is because this type of crop is able to keep producing with minimal input like fertilizer, for example. In lot of cases, smallholders or farmer not fertilize their crop according to the general standard, but this crop still able to producing, still able to have a fruit for harvest. As a note, maybe this is as a note, palm oil came to Indonesia long after other plantation commodity such as coffee, cocoa, rubber, and tea. And the first palm oil in Indonesia is come in 1848, maybe about 100 years ago. And the first plantation was formed in 1911 in North Sumatra. After that, oil palm plantation developed to reach their current area about 60 million hectare in Indonesia. Okay, the studies from all countries where palm oil is grown support that this palm oil is preferred by smallholder because it's more profitable than other crop. So therefore, it is not surprising that oil palm has become the commodity of choice for millions of farmers in Indonesia. Talking about the fulfillment of sustainability principle, smallholder plantation face many challenges, include lack of technical support to meet the sustainability standard of oil palm farming. Then farmers do not have sufficient knowledge of oil palm GAP or good agricultural practices and BMP or best management practices. So the concept are the farmer have low knowledge and skill, resulting in cultivation without referring to GAP or BMP, result in low productivity and low profitability, resulting in extensification without environmental consideration. So this is what happened in smallholder plantation. They need to extend the area of plantation because they need to fulfill their basic need in smallholders. So we will only be able to talk about sustainability if their basic need, the basic need of the family of the planters have been met. It become irrelevant for us to talk about sustainability if the basic needs are still not fulfilled. Basically, the same thing, I think, happen in other part of the world, such as America and Europe. Long ago, exploitation of natural resources also occur in America and Europe. Before it stopped, when their basic needs are met. Now, in Indonesia, sadly, we can say that farmers not already fulfill their basic need. So they will still continue to extend the area to cultivate palm oil because palm oil can give them economic value. So like that. Okay. Now, what is strategy to support the realization of sustainable palm oil plantation? We in Stiper have 3 strategies to support the realization of sustainable palm oil plantation, especially in smallholder. Number 1 is develop a digital-based agriculture extension platform. Number 2 is develop a plantation operational assistance platform. And number 3 is designing appropriate smart technology. I will explain this later. Okay, to build three strategy that I say before, we have conducting research that has been done as a platform knowledge base. All this research is already published in paper, so the audience can search the paper of this research about development of artificial intelligence for VRA, based oil palm fertilization, real-time weed identification, designing disease detection system, development of web-based fertilization recommendation, and literature study about oil palm standard operational procedure. Okay, based on our literature study, if we want the farmer to achieve sustainability, they have to get the knowledge that farmers should know is about 18 points. Start from palm oil agroindustry, palm oil botany, land planning, water and soil conservation, and so on, until laissez-based management practice in marginal soil. The farmer must know this all, so they will achieve sustainability in their operational, in their how they operate the plantation. Okay, talk about agriculture extension platform in the Faculty of Agriculture in Yogyakarta. We collaborate with the Kehati Foundation through the Strengthening Palm Oil Sustainability in Indonesia, or SPOS program that has developed a digital extension platform for oil palm farmer in Indonesia. We call it Sawit Kita. Nah, Sawit Kita, or smallholders assisted with information technology Kehati Institute Yogyakarta, is an information technology based extension platform. In bahasa we call it penyuluhan digital. Is integrated platform consisting of three main pillars. Number one is
Eric, sorry to interrupt. So you still have 3 minutes left.
Okay, thank you. We built this Sawit Kita platform consists of expert system, learning management system, and online chatting. This is the Sawit Kita platform. So there are LMS, or learning management system. We have 21 course and expert system. The expert system is consists bibit kita, OPT kita to identify identification of pests, lahan kita to land suitability class assessment, Verti kita to give farmer fertilizer recommendation, panen kita to record the harvest, income, expense, and other administration related to harvesting, and uang kita, or in this uang kita, there are a financial management system at the level of farmer. Then we also build in this platform AI, or artificial intelligence based camera detection. There are Gulma ID to identification, classification, the weed in palm oil plantation, leaf disease ID, an AI platform to identification and description about oil palm leaf disease. Then finally we have online chatting. In this online chatting, the user can communicate it directly to the expert. They can choose the expert they want to communicate. There are Ispo certification expert, weed management expert, nursery management expert, and so on. So the farmer can directly communicate it, what their problem and what the solution the expert give to them. This program, first we plan to only in 5 area: Paringgian in Central Kalimantan, Sekado in West Kalimantan, Pasir in East Kalimantan, and Mamuju in West Sulawesi. But the realization by Agustus 2022, there are 20 provinces in Indonesia already use our platform. Consists of 2,100 users in all major province producer palm oil producer in Indonesia already use our platform. And there are 3 farmer groups that apply the principle of GAP and BMP using our platform in Paringgian, in Mamuju, and in Pasir. And in international level, this platform is chosen by the SPOC, or Sustainable Palm Oil Committee, in SPOD Innovation Fair in the Netherlands as one of the 3 best idea in the 2022 Sustainable Palm Oil Dialogue. Now, what next? When we talk about what next we will do in Stiper, we will develop of decision support system based on the Sawit Kita platform to supporting the implementation of GAP after palm oil replanting. So the government of Indonesia have program to replanting the smallholders plantation, and we will build a system to support them after the replanting process. And we will also develop of self-assessment and assistance platform to fulfill Ispo certification. So we hope in 2025, when the deadline is come, the smallholder have the ability to fulfill all the requirement in Ispo certification. And finally, we have building or development smallholder oriented mechanization. The mechanization that oriented to smallholder, not to the big company, not in private owned plantation, but to smallholder plantation. And to do that, we hope we can building kerjasama ya, to with all the player in this palm oil plantation, so we can build the palm oil plantation more sustained in the future. Maybe that is all I can share today with you all. Thank you for the time. I'm finished, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, thank you very much, Pak. Thank you very much, Pak Erick Firmansyah, for your presentations. And now we finally go to the last panel speakers. We have here Alice. I don't know. I always call her with Alice, but her complete name is. It's quite difficult, Alice, to mention your name. Zaharichilan Alisa Pavlovna. Is that correct?
Yeah, it is correct.
Okay. Good morning, Alice, in Russian time. Okay, so welcome.
The sun is shining.
Okay, yes. So thank you very much for joining this online mini symposium, Alice. So now time is your, Alice. You have 30 minutes to present. Okay, thank you. Please.
Thank you very much. Hello for everyone. My name is Alice Zaharichilan. I'm a PhD student at Vinogradsky Institute of Microbiology, the Laboratory of Ecology and Biochemical Activity of Microorganisms. I'm happy to join your symposium and would like to show you my study of the hydrolytic enzymes from extremely high alkalophilic archaea Neutronobiforma cellulose-tropha. To begin with, I would like to talk about the relevance of my research. As you already know, various microorganisms are active in biotechnology cycles, in the creation of antibiotics, medicines, fermentation processes, and the production of various enzymes. In the world, there is an increasing demand and large-scale trade in enzymes. The global market for specialty enzymes is expected to reach 14.5 billion by 2027. It should be noted that the volume of enzyme production in Russia is low and extremely narrowly specialized, and there is no industrial base for producing enzymes. The demand is higher than supply. Unfortunately, in 2016, the domestic enzyme market consisted of 95.6 imports. Extremophilic microorganisms have long been used for biotechnological purposes, such as the production of extremozymes. Among the various extremophiles, microorganisms from hypersaline habitats are interested both for finding new organisms, for studying the enzymes, especially hydrolytic ones, or in other words, hydrolysis, or just to study their specification. Among the various hydrolysis cellulosis are targeted in my study. The table shows in the industrial where celluloses are already used. They are textile industry, laundry detergents, pulp and paper industry, juice industry, animal feed industry, and also energy industry. So we can use celluloses in different ways. The research was about to study hydrolytic glycosidase enzymes. Cellulose is an example of the extremely high chlorophylltic archaea, Neutronobiforma cellulose-tropha, ARCL5, isolated and described by Zaharichilan. The strain exhibits high cellulose activity, able to grow on various polymers, cellulose, silan, lignan, beta-glucan, beta-manan, sugars like cellulose and maltose. For example, the illustration on solid medium, which you can see on A, with amorphous cellulose, clearly shows areas of activity around the colonies. The halophilic archaea grow at high salinity, about 4 mol sodium, and high pH values, about 8.59. Under this condition, the amount of biomass is really small. Consequently, following the discovery of the hydrolysis genes, attempts have been made to transfer them to other microorganisms that allow large amounts of biomass to be produced and they target a specific enzyme. The task of the study included searching for genes in Neutronobiforma cellulose-tropha that encode enzymes that degrade cellulose, obtain clones of Escherichia coli and Staphylococcus aureus with insertion of target cellulose genes from Haloarchaea with special tags, HIS 6 tag and strep tag respectively, and I will talk about them later. Obtaining high yields of recombinant enzymes by gene expression, characterization of the properties of recombinant enzymes. The work is being carried out with a framework of a grant from the Russian Science Foundation. It is divided into several stages. First, the genome of the ARCL5 was obtained and analyzed. Next, several dozen genes encoding various hydrolyses were found in the genome. As part of the grant, work was carried out with aid. I was working with only one, but later, of course, I will work with all these genes. The bacterium Escherichia coli and the Archaea halophilus were chosen as model organisms. This is necessary to compare the expression efficiency and properties of the protein depending on the system used. Then transformants were gotten, they tested, selected, and checked. I started my work with ready-made clones. With this, I carried out pre-expression of the gene, purification of the enzyme, and verification of its activity, which I want to talk about. Once we have a good preliminary results, we can move on to full characterization of the enzyme's properties, kinetics, physiology, and structure. And at the end of the work, we can choose possibilities of technological application of this enzyme. Glycosidase genes involved in cellulose degradation belong to different families, and they were found in the genome of the new halophilic archaea. One of these genes was gene, the number which you can see on the slide, encoding an enzyme from the JH5 family, one of the largest families of the nucleic hydrolysis. The family includes enzymes with different activities. When the full genome was analyzed, the gene length was found to be about 3,000. The theoretical molecular weight of the enzyme was calculated to be about 106 kilodaltons. As I mentioned earlier, the endoglucanase gene was cloned in Escherichia coli, plasmid PT46, and Halophilus falcani, plasmid PTA 1392. The PT46 has a lactose promoter and an ampicillin resistance gene. It is induced to be expressed with IPTG, and it has P6 tag. PTA 1392 has a tryptophanase promoter which is activated in the presence of tryptophan, so it must be added to induce overexpression. Strep tag insertion presented in this plasmid. We have tried to use both of these tags in our work. They are most demand. His 6 tag, it is an affinity tag of 6 consecutive histidine residues, and binds specifically to nickel NTA. It has some disadvantages like sensitivity to trees, herpes mops, sodium chloride, and some other reagents. The purification is not high, about 80%, and we need 0 stages to complete the purification. And it mostly used in Escherichia coli. Strep, second tag, is a peptide consisting of 8 amino acids. It binds specifically to main part of streptavidin. It is resistant to sodium chloride and severe proteases, biochemically inert, and purification of efficiency is more than 95% in one stage. It's really good. And it can be used in different organisms. Escherichia coli is mammalian and insect cells, but it's quite more expensive than his 6 tag. Now we are going to the experimental part. In both Escherichia coli and Halopherax volcanic clones, the hydrolytic activity of the enzyme in the soluble protein fraction was checked. The graph shows that the activity is higher for glycosidase from Halopherax volcani on both beta-glucan and lichinan. So we are trying to elaborate our investigation on the endogenous activity cloned into Halopherax volcani. To test the primary endoglucanase activity on Halopherax volcani, cells were grown on rich medium until reaching an optical density 0.6. Overexpression was carried out with 3 millimole of tryptophan at different incubation temperatures, 25, 30, and 42. The cells were then destroyed to obtain a soluble protein fraction. Enzyme activity was checked in carboxymethyl cellulose buffet by concentration of reducing sugars. It appeared that overexpression was most efficient at temperature 30 grade Celsius, and the endoglucanase activity was generally between 50 and 70 microgram per milliliter. In this case, it is rather low activity. It is really small amount, just believe me. It became clear that the existing overexpression protocol needed to be modified. So for this, the original protocol first was modified. You can see the table on the right of the slide, what modifications we have made. As in the previous experiment, after incubation, the cells were destroyed. But now we separated it into 2 fractions, soluble proteins and cell debris. The activity was tested on 3 different substrates: carboxymethyl cellulose, beta-glucan, and lichinan. And the efficiency of the protocol was evaluated. As for the soluble protein fraction, we see the amount of the free substrates of the enzyme activity is significantly high on beta-glucan, followed by lichinan, and the worst in carboxymethyl cellulose. Further, using the original protocol, it can be seen that the activity on beta-glucan was 400, protocol 2 and 3 about 1,300, and protocol 4 about 1,600 microgram per milliliter. The activity is 3-4 times higher in the new protocols than in the original. It should be noted that activity was also detected in the cell residues. Moreover, in the fourth protocol, the activity on beta-glucan is almost the same as the soluble protein fraction in protocol first. This tells us some of the cells are not destroyed, or part of the enzymes remains on the cell residues. The results of the glycosidase activity test showed that an increase in expression time is crucial. It will be logical to continue our work with cleaning the proteins from Halopherex volcani with streptozidine column, but due to some management problems, we have no this column. And not to stop our work, the enzyme activity from Escherichia coli with lower activity were purified in affinity chromatography because we have nickel anti-bond. We measured the activity of the enzyme before and after purification of the column. As expected, there are losses. However, most of the enzymes is lost with elevate. It's really a bad result. So we need to correct our protocols to understand what's going wrong. And not to stop our investigation, we decided to change the plasmid in Halopherex volcani and insert it with plasmid with his 6 stack, and then try to clean it with the column which we have. So the special primer systems were developed. For this primer system, we need to select the temperature for the annealing in project development. A gradient PCR with annealing temperature of primers from 60 to 70 was carried out. And you can see that there are a lot of inappropriate products of PCR from 60 to 44.4 grade of cesium, and from 66.9 TL70 there is no products. So the optimum annealing temperature was 65.6, and after obtaining a sufficient amount of the glycosidase gene product, the next step was to obtain fragments with sticky ends in order to ligate them further with a pure plasmid PTA 1392. The products of the glycosidase gene were restricted. I would like to remind you that the glycosidase gene is 3000 the space long. When we carried out the restriction, we saw that the gene was cut into 2 sections, 1400 and 1600 the space. This was due to the fact that the glycosidase gene contains an additional restriction site CCI1. So the molecular model confirmed this. The chosen recloning pathway turned out to be incorrect. So we need to find another restriction sites to continue our work. So here the resume of the work. The target endoglucanase gene from the strain ERCL5 was successfully cloned into Escherichia coli and Halopherex volcani. Overexpression on the Halopherex volcani clone is better at 30 grade Celsius. Activity on carboxymethylcellulose is extremely low. To obtain more recombinant enzyme from Halopherex volcani in the soluble protein fraction, a new expression protocol was developed. In both clones, the enzyme shows the greatest activity on beta-glucan. The optimal primary annealing temperature for the encoding endoglucanase gene was 65.6, and the elongation time was 180, but it was unfortunately useless. I'd like to say thanks for Leila to inviting me for this conference. I really miss her. So please come to Russia if it would be possible. Thanks for your attention. I will be glad to answer a question if you have, or you can send me questions in my social media. Thanks.
Okay, yeah, thank you very much, Alice, for your interesting presentation. This is about the archaea bacteria, right? You isolated from hypersaline, condition. Okay, all the participants, so we now open for the Q&A session. So if you have a question for three panel speakers here, so please, you can open your mic, or maybe you can only chat in the box. We have a very limited time because now in Friday. Is there any question? Please. Okay, then maybe I have a question for Pa Eric. Yeah, Pa Eric, you were talking about the palm oil plantation and how to cultivate it, right? And also Insiper have build an application, right? And how far the application on that smartphone, I think, is built so far? I mean, so is there any do you apply it, or do you socialize it or already to all the palm oil?
Okay, thank you.
Okay, yes, please.
Ms. Leila, this platform, we call it Sawit Kita, is already socialized, in 4 provinces in Indonesia. Of course, the main palm oil province in Indonesia, in West Kalimantan, in East Kalimantan, or in maybe West Borneo, West Borneo, Central Borneo, and East Borneo, also in Central Sulawesi or Central Celebes. We give the farmer this application free, so they not to buy or to so this is 100% free to use by the farmer.
Oh, okay, that was very interesting. So how does it work? I mean, the farmer have to download it, and then what kind of the information that the farmers get from that application?
Okay, the farmers, of course, have to download it from if they use Android, they can download from Play Store or App Store. If they use iOS, then I say before that in Sawit Kita platform there are 4 main pillar. Number 1 is LMS or Learning Management System. So the farmer can learn about GMP and BMP in that platform. And if the farmer don't want to read the ebook, they can use the expert system. If they want to calculate how much they must fertilize the palm oil crop that have let's say 4 years in a year, they have to fertilize how much. They use the expert system. And if they doesn't want to use expert system, they can directly ask the expert by use the online chatting.
Oh, that's okay. That's it. Thank you very much.
In the Private Kita platform.
Okay, that was very clear, Pak Eric. Thank you very much. Yeah, hope
Thank
everything goes well, and you can develop it maybe more spread out, and then you sell it to all the farmers in Indonesia. Okay.
Yes, we hope so.
Okay, and for Budian, can I ask a question about the urban agriculture? So, is there any differences in nutrient content to some typical of to urban agriculture practical, aeroponic or hydroponic? Is there any differences?
Sorry, can you repeat your
question? Oh yeah. So, is there any differences in nutrient content of the plant? Maybe using urban agricultural, practical.
There are some, I answer. Thank you for your question. So there are some research about that, and actually there is one I ever read one review article which comprise many scientific articles who try to compare that, but the conclusion is there is no different. There is no significant difference between the nutrition content of each technology used.
Oh, I see. Okay, I understand, Budian. Very clear. Terima kasih. And last, maybe I would like to ask to Alice. Okay, Alice, I know that you expert on this Archaebacteria. You are doing this, working on this Archaebacteria for a long time. I know you worked in same laboratory with me when we were in Japan. So the thing very difficult to cultivate and to grow Archaebacteria because they grow specifically only in the very stressed environment. I mean, like the high in temperature, high in saline. So how did you grow it and cultivate that archaea from the hypersaline environment, Alice?
Oh, for Halophyrax volcani, it is easy. It's like
easy.
But in archaea world. But if we are talking about others cultures, other strains, it takes time. Just you put something important salts on the media and just wait that someday they will grow up, or you need to check all the substrates to find which one is more appropriate for this organism.
Oh, I see. So you put the saline with the high concentration, or you put some different various concentration of the saline?
Different salts in different concentration. But of course, if we take the archaea from the saline habitat, we can use natrium sodium about 2 mole. It is really high concentration, and then you check the area from which you take this organism, and you can understand what other salts will be perfect for it. And then you checked all the substrates, polymers, and sugars, and maybe organic acids, but mostly they have not so much the area of the nutrition is really small. They just like only one polymer or maybe 2, not so much.
Okay. So how did you cultivate it for long period? I mean, you put it in the minus 80 degrees or something, and you put some protectant agent, or what?
No protectant agent. The salt is
no. I mean, how did you cultivate it for the long period? I mean, for a long time. Did you keep that bacteria in the minus 80?
Yeah, of course. If we need it, we can.
Okay. So what
With
kind of
glycerol.
agent? So you put in the skim milk or only glycerol?
Only glycerol,
Oh, I
and
see.
it
Okay.
could survive in these conditions.
That's interesting. Okay, Alice. So maybe sometimes I can go to your laboratory and learn from you
It
on
will
how
be very
to.
nice.
Okay, and how to cultivate and growing. Okay, I saw the audience raise hand. It's disappear. Please mention your name. I'm sorry, did I miss it? Okay, maybe canceled. Okay, fine. Okay, to all of the audience, I still have give you a chance to ask a question. Maybe for one participant, please. You can raise hand. No? Okay. Then this is the end of the panel season 2. Thank you very much for all the great speakers for this season 2, and for the Alice and for the speakers. I hope all the participants can get the new insight on sustainable agriculture and also environment. And we prepared the certificate as an appreciation from us to the speakers. But maybe please, the operator, can you share the certificate? Okay, the certificate is our grateful to have all of you. Thank you very much. And Alice, thank you very much. Pak Eric, thank you very much. And Budian, thank you very much. I will send the certificate to your email. Okay. Thank you. Okay. Again, thank you very much again. So all the audiences, so this is the end of the mini symposium 22 on sustainable agriculture and environment. So the master of ceremony now is going to the mosque. So it is the Friday. And thank you very much again for all your willingness to join this mini speakers. I hope we still have another mini symposium on different field area of study, and we can share the idea and also knowledge to all of the speakers and also the audiences. So maybe we can get the opportunity to have the collaboration in the future. Maybe, right? Again, thank you very much. So this is the end of the mini symposium. Thank you very much. Thank you very much again. Good afternoon, all the participants. Okay, bye. Thank you. Ya, terima kasih Bapak dan Ibu, terima kasih para student yang mungkin join di sini. Thank you, Alice.
Terima kasih.
Ya, sama-sama. Thank you, Alice. You can continue. You can continue to sleep again because still in the morning now in Moscow. Okay.
Mr. Kuwarno.
Terima kasih Bu Lela.
Sama-sama, terima kasih.
Matu yuk. Mandul aku.